achievement of BIORIMA. Then we have a session on our contribution, the contribution of BIORIMA to the COVID effort, and then we wrapped up with a discussion about whether we go from here. How can we use the knowledge and the action of the Nanosafety cluster to go further, to address the real-world issue and real-world problem. Okay, with that, I will pass the chair to Lisa. Lisa, are you ready? And you begin the public event. Thank you, Lisa.
Okay, thank you, Lang. Welcome, everybody. I would like to welcome you all by reminding you that now it doesn't look like it, but we are in Turin. This is where we were supposed to be today. And so I will show you just a very quick presentation. So this is where we are, although if you don't, please close your eyes and think that you are here. Imagine we are in Turin. This is where we're supposed to be. Thank you to Enrico Bergamaschi. He already was organizing to be there, but unfortunately we could not be here. And with us, we have external stakeholders. I will just let you very quickly show you who registered for today's meeting, because I think it is a useful information for the speakers. So we have people from the EU, but also people from outside. So welcome also people from farther countries like Argentina, Brazil, Turkey, and Canada. And we have people from different groups of our stakeholders, from the medical, occupational medicine, from the regulatory, from the consultancy, and of course, a lot of interest from research partners. So with this, I will just introduce you to the first speaker of today very quickly, because we don't have much time. In this first session, we will have the presentation of the first of the final results of the work packages. So we start from work package 2 with Magda, if she's here already.
Yes, I am here. Do you hear me?
Yes. Okay. I stop my. So I would like to remind to all speakers that time is important today, because we have to leave time for questions also and for the final discussion, and we should not be late for the next sessions. So please take a look at the screen so you know how many minutes you have. I will let you know when you are 2 minutes from the end.
Okay.
So if you can stop, then we can leave some a minute of questions after your presentation. Otherwise, all the questions will have to be saved for the final part of roundtable discussion.
Okay, so I can start.
Yes.
Okay, do you see my screen?
Yes.
In presentation mode. Okay. Okay, good morning, everybody. Today, I will present the main outcomes from materials, the work package 2, dealing with the selection, production, distribution, and characterization of nanobiomaterials, led by Anna Costa of CNR. Here are the main achievements of work package 2 that we have achieved. So we provide the representative and extensively characterized nanobiomaterials. We supported the standardization of nanobiomaterial production methods. We advanced and improved the current standardized testing procedures of the new class of nanobiomaterials. We provided a wide-ranging characterization in support to risk assessment with safety by design alternatives. And finally, we promoted the development of new ecotoxicological key descriptor for the nanobiomaterials. Here, the list of work package 2 partners. That is a very populated work package with a strong involvement of companies which provided the industrial scale materials. The materials are the key of and the central role, as this a central role in this work package. We selected a relevant set of 20 nanobiomaterials representing real-world needs of industry, regulators, and stakeholders. In order to improve and maximize the impact of the project, we considered different materials in terms of intended use, in terms of composition, in terms of compliance with the European regulations in terms of medical device and ATMP. Then we consider materials in different forms. So we have powders, but we have also suspension and solid materials where the nanoparticles or the nanostructures are embedded on. We consider also industrial scale nanobiomaterials, so with the IRTRL, and lab scale nanobiomaterials provided by university and research center with a lower TRL. The characterization action provided by WRPAC 2 aims to support the risk assessment. Here, the main task that dealt with this characterization action. We collected of all the nanobiomaterials, the basic intrinsic properties by using this characterization technique. Then by means of the task 2.4, we assess the extrinsic properties. So we evaluate the biotransformation of these nanobiomaterials once exposed to relevant fluids that we selected according to the intended use of the nanobiomaterials. Here I reported just 6 relevant fluids, and during the work package we used about 11 fluids. Then task 2.6 was focused to assess the surface reactivity of the nanomaterials alone and also when exposed to the relevant fluids by applying technique like, for example, EPR, or dissolution by ICP, cyclic voltammetry, and also by using methods inspired and taken by the catalysis field. The main aim of this characterization, widespread action, was to correlate the biotransformation with the nanobiomaterials properties to improve the predictive power of in vitro tests and also to correlate the cellular data with the nanobiomaterial properties. Here a list of the materials that we tested, exposing them to the relevant fluids. We considered lab scale, industrial and benchmark materials for a sum of 11 nanobiomaterials dispersed in 11 media. This is a task and the work performed by all these partners, and the results are into deliverable 2.5. Here are some brief results taken from the activity performed particularly by the University of Torino that explored the biotransformation of nanomaterials exposed to the gastrointestinal tract, then monitoring the size distribution, data potential, so the colloidal properties of the nanomaterial, the surface reactivity, and the morphology. They exposed the materials to fluids typical of the gastrointestinal tract, saliva, gastric fluid, and duodenal fluid. Just an example on the solid lipid nanoparticles that are particles provided by the company Nanovector for food supplement. And here are the results of the evolution of the hydrodynamic nanometer exposed to all these fluids. We can see that we have a variation, and at the end, when we arrive to the duodenal fluid of the digestion, you have a split of the peak with the coarsening of the particles, but also with the appearance of the smaller particle, indicating the starting of the digestion process, as then proved by the TEM analysis. The materials have been also tested in terms of surface reactivity by means of EPR technique, EPR characterization. Here are some examples of testing of carbon nanoparticles, magnetite provided by Colorobia, coated by PG and PLGA, and hydroxyapatite doped with iron. We see that the EPR signal is changed if we expose the materials to a cell fluid, in this case, DMEM. For magnetite and hydroxyapatite doped with iron, we see that the surface reactivity is modified and reduced when exposed to DMEM due to the formation of a biocorona. For carbon nanoparticle, instead, we didn't see any variation of surface reactivity to the presence of DMEM. For other materials, like, for example, hydroxyapatite doped with titanium, we didn't see any kind of surface reactivity, any kind of signal, both in dark and illuminated conditions. The activity developed by CSIC was addressed to monitor the oxidative reactions and the oxidative capacity of the nanobiomaterials. And to do that, they developed new methods inspired by the catalysis field. They put in contact the nanobiomaterials with probe molecules that can be oxidized by the nanobiomaterials and then treated in temperature. Depending on the kind of oxidative products that they detected by Raman spectroscopy, they can rank the oxidative capacity of the explored nanobiomaterials. And in addition, they can also tell how many reactive sites we have, and also the role of different active sites by applying a method called temperature programmed surface reaction that starting from a methanol chemisorbed on the nanobiomaterials and then treated following the process, can identify the different reactive site of the nanomaterial, acidic, basic, and redox, depending on the kind of product that they detected. So these methods make available the study of the surface reactive sites of the nanobiomaterials in terms of number, distribution, and also reactivity of them. Dissolution tests in relevant media have been performed on many materials. The method that we developed is here schematized. So we put in contact the materials with the relevant fluids using different concentration for different time points at an incubation temperature that is 37 degrees C for toxicological fluid and 20 degrees C for echo fluid. Then we proceed with centrifugation and separation of nanoparticles from the ionic fraction that is typically assessed by and analyzed by ECP in order to measure the dissolution. Here some brief results of magnetite provided by Colorobia. This is the colloidal data in water. And here the results of the colloidal behavior when exposed to cell media, RPMI and RPMI plus FBS. We can see that to the stronger and the high ionic strength of the RPMI, we had a strong aggregation of the particles. With the introduction of FBS, we have instead a dispersion effect with the lower hydrodynamic diameter. Passing to dissolution, we can see that the presence of FBS slightly reduce the dissolution of the material that we test at different concentration for different time points. We also observed that for the first 24 hours, the dissolution are very similar, quite similar for all the concentration, indicating a sort of equilibrium between solid and liquid phase. while increasing the time, we have an increased dissolution. Passing to another fluid, at the phagolysosomal simulated fluid, we have a different behavior. We have also, in this case, a strong agglomeration due to the high ionic strength of the media. But in this case, we have a lower pH because this is a typical acidic fluid. And what we see is that in this case, the dissolution is higher for all the concentration, and we didn't see any kind of equilibrium. So we have different dissolution curves confirming time and concentration dependency. Okay. We also developed safety by design case study within work package 2. And we consider and we developed 3 main safety by design case study. The first one dealing with the polylactic acid fibers doped with silver sigma provided by the electrospinning company. And we proposed as alternative design the substitution of the silver with silver cellulose, so a silver nucleated in a cellulose matrix by means of eco-friendly synthesis, and providing the fibers doped with this kind of coated silver for wound dressing technology. The second case study deals with titania for sunscreen application. When we proposed as alternative material to typical titanium nanoparticle, hydroxyapatite doped with titanium embedded in alginate biopolymers. And finally, we compared in terms of reactivity magnetite of sigma Aldrich, so a naked and basic materials with the magnetite coated with the bioresorbable coating of PG-PLGA, and the hydroxyapatite doped with iron, which correspond to a totally bioresorbable structures. These materials are, as intended, use theranostic application. The development of safety by design case study implied a strong collaboration between work package 2 and work package 4 dealing with the ecotoxicological characterization. So work package 2 provided the materials and prepared the material and all the characterization in support of the risk assessments, with providing all the data on how the materials transform in the relevant fluids for use stage and for the end of life. So media taken from the ecotoxicological field. And considering the activity in collaboration with Work Package 4, they provided all the data in terms of toxicological and ecotoxicological assessment for the case study of silver in PLA fibers for wound dressing. We collaborated with the Journeyum Research, which provided data by means of the open flow microperfusion equipment, providing data from ex vivo and in vivo experiments in order to provide the data on pharmacokinetic of silver, the biological fate, and the toxicological effect on skin. Considering the environmental impact, we collaborated with the University of Aveiro, which assessed the silver interaction and the ecotoxicological effect with the soil model species.
Magda, you are wrapping up? Okay,
just very brief. I finished the time, yes?
Yes, 1 more minute.
Okay, just concerning the functional properties, we assess the functional properties, so the antibacterial properties of the modified version that we proposed, the silver cellulose, that showed improved performance with respect to the naked silver sigma, that is the basic materials. And this means that we have an increased functionality, so we can add less material to have the same performances. Okay, I can skip this slide. Just 2 words about the other material that we proposed as safety by design alternative, that is the hydroxyapatite doped with titania that showed high reflectance, particularly in the UVB, UVA portion of the spectrum, that is very useful for sunscreen application. And at the same time, this material did not show any kind of photocatalytic activity that instead is shown by typical titania used for sunscreen application. All the methods and all the data used and applied by Work Package 2 are enclosed in deliverable 2.9 and have been translated in the fact sheets of deliverable 6.3. Okay, I finished. Thank you for your attentions, and these are the contacts, the main contacts of Work Package 2.
Okay, thank you, Magda. I think we can take one quick question if anybody has one question for Magda. Otherwise, we can go straight to the next presentation. So no question now for Magda, then at the end we will have a roundtable discussion and all the themes will come together.
Okay, thank you.
Thank you, Magda. Good job. So we have Bernd Nowack now presenting the results from Work Package 3, which was focused on exposure studies.
So welcome everybody. I hope you can see the presentation mode.
Yes, we can.
Okay, perfect. Thank you. So I will show you results from Work Package 3, which was dealing with the exposure to nanobiomaterials. So the main results from Work Package 3 are, I need to move this here. So we have what we identified are release and exposure scenarios for nanobiomaterials. We then characterized release of nanobiomaterials from materials. We also specifically developed methods for evaluating nanobiomaterial release from implants. We then looked at environmental release and exposure to nanomaterials based on modeling. And then an important part, looking at exposure and biomonitoring in occupational situations. So the important for our approach was that we look at the complete life cycle. So it means a nanobiomaterial, there's not only exposure to the patient, what we normally would consider, but also these nanobiomaterials, they have a complete life cycle. They're produced, there's the manufacturing of a product, there is then the use in the patient, but there's also the professional user, there is some end of life. So there may be workers exposed, there may be releases to the environment. And our work package looked at this exposure from an overall perspective. So on the modeling side, we developed material flow models to follow the flows of these nanomaterials from production to administration in the body. Here, especially to look at the fate, what is biodegradation, accumulation, and especially the excretion. If it's degraded, stays in the body, the way the nanobiomaterial can be completely transformed. But if it's excreted, it ends in wastewater, from where then finally it may end up in the environment. There may also be releases during manufacturing to the environment or into technical compartments. So we developed the material flow analysis model so that we can track the flows of nanomaterials, and we applied it to several materials. And with this, we were able to predict environmental concentrations in water and soil that then could be used for an environmental risk assessment. These predictions are based on some scenarios. We also then to some extent looked at further environmental fate. So we added a small model on the further environmental fate in the environment after release to some environmental compartments. So on the release side, our focus was on unintentional release during production and manufacturing of products containing nanobiomaterials. And in these situations, the release scenario depends on the material properties. So what is actually the material that contains your nanobiomaterial and the process that this material undergoes. And what we focused here were lab simulations of mechanical treatments. For example, here, this test block collagen with the magnesium hydroxyapatite nanoparticles. And now there's a drilling operation producing some dust. This inlet tube here is collecting the air. And then there were some characterization of the particles released into air. And this would be what the workers working with these materials would be exposed to. So the characterization of the particle size distribution here, the nanoparticulate size, but it's mainly in these operations that there are large particles produced and not single nanoparticles. And this is always accompanied by TEM investigations looking at the materials before and after these operations. A specific part of this work was to develop methods to look at release from implants, where there was a method developed and validated to test exposure and toxicity of implants in synthetic synovial fluid. And there was a new biological system developed to assess then the biological impact of the wear particles, so that these small particles that are generated. So at the end, it was a proof of principle established for a parallel exposure and hazard testing. So on the one side, produce a braided particle from such implants, and on the other side, test the hazard for both the particulate and the dissolved phase, because many of these implants, they consist of metals that can both have a particulate and a dissolved phase. So this part of the work is on the way to standardization. There, at the moment, it is on the stage of a workshop agreement, and this is one of the steps towards a CEN standard. Two parts: the method to produce the wear particles from these implants, and then this method, this tiered method to evaluate the biological impacts. Then on the exposure monitoring side, an important part in work Patrick 3 here, it was first to develop strategies how to monitor exposure to nanobiomaterials in an occupational setting. So again, the focus here was on the worker handling materials containing nanobiomaterials. There was first a strategy developed, and then it was validated in a field campaign where we looked at exposure and also did a biomonitoring study. So the framework for this field campaign follows the standard tiered procedure for exposure assessment, where the first tier, it's information gathering from possible situations using questionnaires sent to companies, some scoping visits, seeing if there is potential exposure to nanobiomaterials. Then there was a tier 2, a basic exposure assessment using handheld portable devices to check for release of particles into air. So the focus was here occupational exposure through the air, as the lung is the most important entry organ in such situations. So, and based then on these measurements, it was decided that there were 2 situations that were very promising, and especially one was investigated in more detail in this tier 3 and the expert exposure assessment with standard analytical equipment that is used to characterize airborne exposure to particles. So this was done at the company producing dental prostheses, they worked with a material that is as close to nanobiomaterial as possible. So it contains a nano-sized fraction. It is one of the ceramics used to produce the third teeth. So, and there was then this exposure monitoring campaign with all the instruments that you see here. Here's with the sampling here close to a 3D printer, or here sampling close to this machining, this ceramics that is then becomes here the third tooth. So definitely a situation where there is possible release of particles into the air. So there were measurements conducted looking at particle numbers close to the worker, but also far away in the room. So to get the idea of the background contamination of the air with particles, but also in a way, the influence of certain actions on release of particles. There was also offline analysis of particles collected and then identification by SEM on the elements in these particles and the detection of the different metals and other elements that were used in the processes in this company. So the conclusions from this exposure monitoring was that there was a high aerosol background throughout the company. There was then some specific release doing specific actions, but it was all its non-intentional release due to different processes, but these are not in a way exclusively nanobiomaterials that are released, but nanoparticles produced during these mainly mechanical actions. So it's a mixture of, in a way, ultrafine particles, fine particles, and some identification of these materials, these ceramic particles that were present in some of the materials that these workers handled. This study was then complemented by a biological monitoring where the objective was to identify relevant biomarkers for monitoring exposure and early health effects. So looking at biomarkers of exposure and biomarkers of early effect. So from the exposure monitoring, we knew there is some exposure of these workers to particles in air, particles containing some metals that are known to have potential health effects. So the procedure of this biomonitoring was, of course, a detailed questionnaire about the workers that participated in this study. Then there was a biological sampling of this exhaled breath condensate, urine, and blood before and after work. And then there was analysis of biomarkers of exposure, so various metals in this exhaled breath condensate and metals in urine, metals that were found to be present in these particles in air, and then several biomarkers of early effects, biomarkers for oxidative stress, inflammation, and fibrosis. So just a very short summary or conclusions from the biomonitoring, that there were in principle no differences in the level of biomarkers of exposure in workers compared to a control group not exposed to these particles in this company. There was one biomarker, there was a significant increase, this interleukin-1 beta in the exhaled breath condensate, but we have to be really careful with these, with any conclusions, because there was just a very low number of subjects. This was during the COVID period, so it was very difficult. There were a lot of delays in carrying out this biomonitoring study. So I think as work package leader, I can be really happy that at the end they managed, even in these difficult times, to both do the, in a way, the exposure monitoring and the biomonitoring within the time frame of the project. There were some delays, but at the end we managed to do this. One of the first, in a way, complete exposure monitoring for nanobiomaterials. So the general conclusions from work package 3, we can definitely say that we achieved a significant advancement in the nanobiomaterial exposure assessment on the side of modeling release assessment, but also exposure. And very important, and I think this is what we highlighted, it's not only about exposure of patients when we talk about nanobiomaterials. There can be environmental release. This depends on the application. There can be release from products. Again, it depends what are the actions you do with a certain material. And we also need to consider there is the potential for occupational exposure to workers. And within our work package, we performed the first biomonitoring study as a baseline now for future work. So, and with this short out, in a way, outlook from work package 3, and thank you for your attention.
Thank you very much, Bernd, for this very clear explanation of the results and the conclusions from these exposure studies from BIORIMA. It was very clear. Does anybody have any specific questions now for Bernd before we get to then the final discussion, and then that's where people can ask cross questions, let's say. So if there is no urgent and impelling need to understand something of your presentation, we can go on. Thank you, Bernd. The next presentation is about work package 4, and for the followers of BIORIMA, you know that work package 4 had 2 souls. It's about the hazard studies, both from the human and also from the environmental point of view. So we have now Bengt Fadeel, the leader of our package 4, leading this presentation. I leave the word to you, Bengt.
Okay, thank you very much, Lisa, and good morning, everyone. I'm going to give a 10-minute presentation, and this will be followed by another 10-minute presentation from Janeck, who will cover the environmental part of the work. But in principle, work package 4 comprises both human health and environmental studies. And it is a very large work package. Basically, half of the BIORIMA project is represented in work package 4. So I'm not really going to attempt an exhaustive description of all the work, but I thought I would give some highlights in the presentation today. First of all, the aim of our work has been to develop reliable and robust test methods, specifically to evaluate the adverse or potential adverse health effects of so-called nanobiomaterials. And by this we mean nanomaterials that have an intended use in medical devices or advanced therapy medicinal products. And not only to look at the potential human health effects, but also to develop test methods and schemes to evaluate potential environmental effects. I'd like to focus today in particular on iron oxide nanoparticles, and just to give a short introduction to this topic. In this slide, I summarized some previously published work on one particular type of superparamagnetic iron oxide nanoparticle, which is known as ferromoxitol. This is a carbohydrate-coated iron oxide, and it has been FDA approved for as a supplement in iron deficiency. What I'm showing you here in this slide on the left-hand side is a result of a clinical study in which these nanoparticles were used as contrast agents for imaging of atherosclerotic plaque. I'm not going to go into much detail here, but for more details, I refer you to the review article mentioned at the bottom of this slide. That we wrote actually as part of the BIORIMA project. On the right-hand side is a preclinical study. This is a paper published several years ago by other authors, where the same nanoparticle, this previously FDA-approved particle, was used in a model, in several cancer models actually, but in this case, a model of liver metastases. And interestingly, these particles did have a strong effect, but not necessarily on the tumor cells. Actually, it turned out that the particles were reprogramming or polarizing macrophages within the tumors, and this was driving the response. This is one example of an iron oxide nanoparticle. Now in BIORIMA, we have studied a different variety of magnetite, and as you can see at the top of this table, the table was of course prepared by work package 2, and it covers all the materials that we have been screening and testing in this project. But at the top of the table there, you see the arrow on the left-hand side pointing to the iron oxide PEG PLGA material from the company Colorobia. And you can see highlighted in blue in the middle of that table the intended use or applications for this material, which is both in drug delivery and in imaging. So essentially, in principle, a theranostic type of material. There is another arrow further down, and this points towards reference or benchmark materials, which I prefer really to call them. And these are largely materials from the nanomaterial repository at the JRC. And these are the traditional materials that we and others have been testing in many previous projects, including the Marina project, which really is the precursor of BIORIMA. Now, on the right-hand side are some of our unpublished data from our lab showing that these iron oxide particles are non-cytotoxic, both in a macrophage model at the top and in a neutrophil model at the bottom. So that's good news, obviously. But in BIORIMA, we were, in our work package, we were interested in identifying, let's say, more sensitive endpoints. This assay is an LDH release assay, and it really is an assay that reports the rupture of the plasma membrane with release of the LDH enzyme. Now, in short, we focus antioxidant response in one of our joint studies in work package 4. This figure is rather busy, but it's from an excellent review by Helmut Sees and Dean Jones on redox biology in general. And perhaps you can see in this figure, I don't know if I can point, I can give it a try. We have NRF2 here, which is a transcription factor which drives the antioxidant response. And NRF2 is a key player in this antioxidant protective response in cells. So in work package 4, we decided to use an NRF2 reporter cell line and to do a so-called round robin or interlaboratory study. And these cells were provided to us through a material transfer agreement with the company Biodetection Systems. And this company has, first of all, I have no affiliations or ties with the company whatsoever. But just to say that this company has previously developed other reporter assays, which are now adopted by the OECD and used for testing of estrogenic and androgenic activity of chemicals. The test materials were the iron oxide PEG PLGA versus the two benchmark materials, titania and silver. And 8 partners in work package 4 were involved in the testing, and we also collaborated, of course, with work package 2 in this study. I'm not going to show you the results, but just to say that this round robin was comprised of 3 steps. First, we did have a series of two tutorials with the company to really make sure that everyone knew exactly how to conduct the assay. And then we all went off and did our testing of the 3 different particles and additional chemical controls, I should say. And finally, the Wageningen University that coordinated this study has collected the data and performed the statistical analysis. Overall, what we found is that the iron oxide triggered a rather modest induction of the antioxidant response, whereas the silver particles triggered a strong dose-dependent response, and titania, as expected, actually was shown to not trigger any response. And this was largely concordant across all the participating labs. This is what we would then refer to as a pre-validation study. It is, after all, a rather small round robin with 8 participating laboratories, but we showed in principle that the assay works, and this could be therefore useful in terms of developing future standards to address the antioxidant response. I'd also like to say just a few words about in vivo testing, since this has been a very significant part of the work during the last period and during this actually during this pandemic period. And I have to say that it is quite impressive, really, at least in my mind, that the partners listed here managed to pull this off despite a severe lockdown in many cases. The partners are listed at the bottom. So we had 3 European partners and 3 international partners involved in these in vivo studies using rodent models, both mice and rats, but also a porcine model. And there were also additional partners involved performing ex vivo analyses of tissues and samples from these animal studies. And again, we collaborated, of course, with Work Package 2. So the overall rationale, obviously, as stated at the top of this slide, is that we have been screening a large number of materials in vitro using a range of models, but we then selected a few materials for in vivo testing. And again, we focused here in particular on the iron oxide PEG PLGA and other spions, as these are important representative nanobiomaterials. And we did spend a great deal of time to ensure that the materials were endotoxin free and so forth. I'd like to give just one example. This is work taken from Trinity College Dublin, the partner coordinating this in vivo, all the in vivo work. And here the colleagues have tested 2 different spions using 3 different concentrations and an IV route of administration. The bottom line is that there are no gross abnormalities in the key organs shown here. The arrows in the lung sections point to some infiltrate of eosinophils and so on, but no gross abnormalities. Trinity College Dublin partners also checked for iron accumulation, first using the classical Prussian blue staining, which you see in the images on the left-hand side. And this was corroborated using ICP-MS, which is, of course, a very sensitive method, as you can see on the right-hand side in the 2 graphs. So while iron is present in organs, in particular liver and spleen, to some extent in the lungs, the levels are very, very low, and the authors here concluded that these are within tolerable limits. As an overall summary, I have to say that the Work Package 4 partners, there are again 18 partners in this work package, have generated a huge amount of data, both with respect to classical benchmark materials and also new and more advanced nanobiomaterials using both in vitro and in vivo models. And I covered some of the human health aspects, and Janeck will shortly cover the environmental aspects. All of this data and also the underlying protocols are available through the BIORIMA database. And based on this work, we may suggest modifications of existing guidelines, and we may also suggest new assays for further testing. So with this, I'd like to stop and give the word to Janeck, who will cover the environmental part of Work Package 4. So please, Janeck.
Thank you very much, Bengt. Can you hear me?
Yes.
Sure, perfect. Thank you very much. I'm going to share my screen now. So just you can see my screen now.
It's coming, yes.
Perfect, okay. Thank you for a very nice overview. And indeed, it is a very big work package. I'm putting it on. Good, okay. I will tell you what happened in the environmental part of the work package. And we have dealt with the same material as Bengt has outlined and that Magda outlined for Work Package 2. So we have coordinated work with these, and the 4 partners that were in here were Monica, who was leading this work in the environmental part of this work package, Kerstin Hundbrink and Louisa, and myself here. And what we did was, I can say that BIORIMA did not stand still in any in any moment, even with the COVID. We try to follow the surrounding movements, and like everybody else knows, there is a high focus now on the Green Deal that came up a couple of years ago, and on sustainability, long-term sustainability, chemical safety by design, sustainable by design. So we try to incorporate all of these things along the way, as did Bengt and the other partners also did. So I think that was a very fruitful process and a very nice movement forward. So what we did within this work package was that we moved forward with evaluating the general challenges that are within the OECD guidelines. How do they perform, and what is the issues that would be coming up with nanobiomaterials or medical products? What would the main issues that we would have to deal with? We try to move forward in a way that we try to move from a black box to an open box, in the sense of that we try to make the test systems tell us something about the mechanisms of actions, not only on a biological minute level inside the cells, but also on a system level. So what can we do from one material to another material? So we would use that for material testing, but we also use that to make safer by design development in the future. So you know something about what happens on the mechanistic level, then you can design new materials that are more safe or safer. And we evaluated these guidelines, and we developed new guidelines. We extended the guidelines that are already there. So they're all based on the full life cycle test. We extended life duration, bioaccumulation testing, and mechanism for actions like omics studies. And then, of course, for all the studies, we tried to evaluate the variation, whether that was different from the current standards. So that was the way forward here. So the evaluation of the OECD guideline, what are the changes that we need to have? And we went through the list of OECD guidelines, and they more or less are built up the same way for the environmental part. So you have the principle of the test, you have the text acceptance, and so on. So we evaluated the different steps, where is needed, where's the change needed, where there's no change needed, how can we adapt it, what can we suggest for that? And then we published a paper on this, so we were sure that we were on the basics. Then we evaluated the current testing systems that are out there in the literature, the open literature, scientific literature, what has been done on nanomaterials, which is related to nanobiomaterials, which tests are out there, which compounds have been done. As you can see, for example, in the A to the left, there's mainly earthworm studies that has been performed, some encotrait studies. If you see on B, you can see there's a high degree of silver testing, some zinc testing, and so on. We evaluated that before, so we had a baseline to go for. Then we developed the various methods. I'm gonna go through now a list of maybe 10 different progress we did. So one is we did a standard, which is on the left-hand side. You can see there's the standard. This is the standard OECD test guideline, 28 days. We evaluated one of the, all the materials, but one material here is the iron PEG, for example. Then we made an extension, 56 days. And on the right-hand side, the full life cycle test. This is actually not the full life cycle test, but this is a representative of it. We studied the individual step in the growth of the organism. How did they develop after 7, 14, 28, and 46 days, 56 days actually. And then we see whether we can tell something about which steps in a life cycle is mostly affected. And there was differences for different compounds. We also tested do they live longer and shorter in various cases. We also tested bioaccumulation of the different compounds. Here, for example, for the silver, we tested the bioaccumulation of silver compared to the nanoform, the iron compared to nanoform. And in the lower right corner, if you're fast, you can see that the nanoform seems to accumulate for a longer time, although this may depend on the individual materials. We also did some multi-generation testing. So one of the things for sustainability in the environment is, of course, that things stays in the environment for a long time, and organisms are often exposed to more than one generation. And what happens in the second generation, or is there a transfer effect? So we tested up to 5 generations, also with generations coming from contaminated into clean areas, again, to see whether they're persistent. We make some omic studies, mechanism of actions. We try to develop some AOPs, adverse outcome pathway, which is a mechanistic understanding of going from the gene expression over the proteins to the metabolomes, and then link it up to the population responses, something like effect on reproduction and so on. We also did an integrated study between the partners where we studied how would one medical product or medicine come into the environment via the sewage sludge, being in the sewage sludge, being in the treatment water, then the sewage sludge would be added to the soil, whereas the sewage water will go into the river. And then we studied the organisms that would be in the soil receiving this sludge, and then we studied also the organisms in the river and seeing how they would be affected. And we made this integrated study, and this study was made on gold nanoparticles. So that was also very neat. It's also been published now. On the microbial, we studied the microbial, a lot of different microbial parameters, potential ammonia oxidation, which is very important for the nitrogen cycling, exoenzymes, which are enzymes that are outside, the organic degradation on degradation of organic compounds, which are on the right-hand side. And a novel technique also, because EFSA and other organizations are asking for more information on the diversity of microorganisms. So we studied also the composition of the microbial community within the soil and the sludge. But here is an overview of that, and you make a sequencing, DNA sequencing, and then you see whether there's any changes, and then you can plot that on the right-hand side with the multivariate statistics. We also adapted the in vitro test systems with the fish guidelines, fish cell guidelines. We extended it from 24 to 96 wells. And we did very nice work also on the short-term to long-term, increasing the exposure duration from 24 hours up to 14 days and then up to 28 days, seeing the difference in toxicity and seeing that there is a potential recovery after a time. As you can see on the right-hand side with the graph, there's some of the graphs have a small slope up to at the end, and that is a kind of recovery in this sense here. And then, so the outcome, I know this was a very fast overview, and there are more tests being developed or have been developed, with just an outline here. But one of the things we did is that we tried to make a lot of OCD test systems updated to a longer term, more sustainable perspective, so they can be used for nanobiomaterials. We tested the reliability and relevance of these for the OCD system. And then, of course, this is targeted for the Green Deal and chemical safety strategy, and so on in the environmental perspective. That was my words. I hope you could follow. Thank you very much. Let me know if there's any questions, or Bengt, you take.
Yep, that's great, Janeck. Thank you. So I guess over to you, Lisa.
Yes, thank you both. Thank you all the speakers for so far. You have been great in timing. And I think it's very difficult to wrap up all these data in just a short time. So if there are no specific questions for these two parts, we can go and start the, I guess. I don't see anything even in the chat, so we can start the coffee break.
Lisa, yes, sorry.
Yes. Oh, Flavia, good morning.
Flavia here, good morning.
From Istituto Superiore di Sanità, which is a regulatory research stakeholder of BIORIMA. She has been following us since the beginning of the project. Thank you, Flavia, for being here.
Thank you for the invitation. I would like, first of all, to give many congratulations for the project, many results achieved, still considering the difficult period that we are living. So my congratulations. I have just a little question to Bengt. You spoke about the data, in vitro data used as screening, a pre-screening rights of these bio-nanomaterials, and all the data BioRIMA produced are in a database of the project. So my question is, this data will be public or not? Because I know there is many difficulties in making this data public before publication, of course. But my question is related to the work is in course in many European projects to produce fair data. And in this respect, it would be very interesting to have the possibility to use your data. I imagine you produced a lot of data in many different fields.
Right. So,
Ben, you want me to answer this question? It's Lang here. Should I do it? Hello. One of the things that we did mention is the BioRIMA database. So all the data, the database, all the data and the SOP and everything are collected and put it in the eNanoMapper format. So after the project is done, of course, there is a caveat on the quarantine period for the partner to publish their data. Then the data will be made public, and we will upload everything to the eNanoMapper people, and upon making a request, you'll be able to access all this data. This is one part of the BioRIMA, which is the legacy part. So we didn't mention it in our project meeting yesterday, but it has always been the work that has been performed. So does that answer your question, Flavia?
Thank you very much, Lang, perfectly. And I am very happy to know that the database you use is eNanoMapper, because in Go4Nano, we are trying to use many data for fair approach using this database. So I think it's very important. Thank you.
Yeah.
Great. There is no more question now. We can start the coffee break.
Coffee break.
Please come back. Please come back before in 5 minutes, so we can start at 11:10 sharp.
Yeah. Une recommandation pour le oui? Oui.
Okay. We start at 11:10 sharp. Thank you.
Generate physical, chemical, and hazard information for the hazard identification process in a cost-efficient and structured manner. The decision support system also uses advanced models to perform dose response assessment and both occupational and environmental exposure assessment along the life cycles of medical applications. And these are models which were specifically developed in BioRIMA, and they were connected to the system to enable assessment of fate and exposure. And the focus is always occupational and environmental, because in parallel to the safety for patients, we're really focusing on trying to assess the risks of the nanobiomaterials also to workers and medical staff exposed to the materials in the use phase. And also on the implications of these materials ending up in the different environmental compartments. So these models, they're important standalone results of BioRIMA, and they were included in our system, where we chain them to risk assessment algorithms. The algorithms use results of the models and data sets that can be either manually imported to the system or derived from databases to perform quantitative occupational and environmental risk assessment for specific scenarios. The assessment is always quantitative. It can be deterministic or probabilistic, and there is always uncertainty analysis. This has proven to be really important for the interpretation of the results and to better inform risk management decisions or regulatory decisions. So the uncertainty analysis is an important part of it. Then the system also helps you to identify if risks are acceptable or not. If they are not acceptable, then it guides you through selecting suitable risk management measures based on information on the efficacy and the cost of the measures. And all this information is available through the SL database, which was kindly provided by TNO in the Netherlands, and this database is regularly updated with new information on risk management measures. As Alex already hinted, the DSS is now fully functional, and it's publicly available via this link. The DSS was also demonstrated in some key BIORIMA case studies, and for each of them, peer-reviewed publication is available. With this, I leave Alex to show you the real system, and then Virginia will say a few words, only a few words about the case studies, because our time is very limited today. Alex, please. And sorry for the technical problem again.
Thanks, Daniel. So let's share my screen. Okay, so as we were starting to see beforehand, the BIORIMA decision support system is included in the SANS infrastructure. You can select it from this initial interface. Once you start the BIORIMA DSS, you have two possibilities available: the application of IATAs and the risk assessment. Usually you first go through the IATAs to understand if risk assessment is necessary in many cases. So if we enter the IATA, this brings us to the specific component. In this case, you can create an IATA for a specific material, for example, Magamite in this case. You can specify the components of this material. You might go also through a classification. That's not the aim of this presentation, but you can perform basically IATAs. So I defined two scenarios, generic scenarios, one for the oral route and the other one for the inhalation. And then I can apply the IATA to the Magamite for the two scenarios. So as you can see here, we have these two parts, the inhalation and the oral, and inside we have the different IATAs. If we click a specific IATA, we can start it. So an interesting thing is we can take a look at the full scheme of the IATA so that you can see the complete flow chart of the different decision nodes included in this IATA. And then we can start its application. So the first, and it is a series of questions you should answer to. The first question obviously is already answered because we are in the oral scenario. And so the route is oral, but then we have to decide which is the purpose of this application. It can be safe by design or it can be regulatory. Let's suppose we go for a regulatory setting. Then we have a series also of specific questions. Each question presents some introductory text, the possible essays that you can apply to answer the question, and a set of guidelines to be followed. Also, this can be performed at different tier levels. Each tier level implies a higher precision in the assays that are applied and in the results. And it's up to you deciding which tier you want to stay in. And you can go on and reply, for example, that this is a quickly dissolving nanoform. And then you have questions related to cytotoxicity. And again, you can see the different assays that can be applied. So you can decide that it's not inducing cytotoxicity. Then you move to surface reactivity. And this might be a yes. And the tool goes on like this. So you can continue to answer these questions until you get to the last one. Sorry. And so, for example, in here, because we can have, we exhibit some surface reactivity, we should be performing a full risk assessment as we are in a regulatory setting. So by following the link, we go back to the other part of the tool, so the risk assessment part, where you can have several projects. So you can see the Magemite product I was working with in the IATA is still here, and we can also see other two case studies that were shared with me by Virginia. And Virginia will be presenting the case studies after this short introduction to the software tool. If we enter, for example, the ingestion case study, you can see here we have this one. This is an assessment context. It means a way to develop this product. So in this case, this is the activity going on. So the dispersion of magnetite, NP is coated with PGA and whatever. You can see we have a description of the nanomaterial used. And here we can also decide what should be assessed by the system. So you see that here only the occupational part was selected. The synthesis, product, manufacturing and use life cycle stages were selected. And inside the life cycle stages, only the inadvertent oral route of exposure was selected. And if we enter this assessment context, you can see the system allows to perform a risk assessment or risk control procedures in the risk assessment part. In this case, Virginia already input the data, so we already see the results here. But you can see on this left side menu that the environmental part is disabled because we only selected the occupational one in the assessment tree beforehand. And in the occupational part, we can assess exposure and hazard. If we go for exposure, for example, you can see we have 3 rows that are related to the aspects we selected before. So it's always the inadvertent oral route of exposure for the different life cycle stages of synthesis, product manufacturing and use. What you should input is exposure levels, and this can be done in a probabilistic, deterministic, or you can even say there is a negligible exposure without even specifying values. In this case, we went for the probabilistic exposure, which is the most detailed way to express exposure. We've been inputting lower and upper confidence limit for exposure, and this can be done manually, or you can derive it by applying a model. So in this case, you can see this has been input by a model. And the tool in general works like this. You can always find this green play button that brings you to the model, in this case, the IEAT model. And this is how a model looks like. So up here, we have this brief recap of previous evaluated results. And this is here because we already applied the model beforehand. And here you can see all the inputs that are needed for this model to run. All the models provide default values for the inputs, and the user can change according to the specific case and rerun the model once the data has been changed. Also interesting is for every model you can find a reference to the model supplier. So these models are mainly from project partners, and some of them has been developed directly by us. The same thing applies for the hazard part. Again, only inadvertent oral and the different life cycle stages are reported here. Again, you can input in this case, the DINEL lower and upper confidence limits. In this case, you can run this dose response model that is based on PROAST. I'm not going to open that, but it's quite similar to so the interface is the same. You input the data and you run the model and you get the results. Then after you input all your data or launch the different models, you can take a look at the results. You can decide what you want to see here. So synthesis, in this case, the only difference is in the life cycle stage because the root is always stat 1 and the risk is always occupational. This is the distribution curve for the exposure derived by the model. We have the chart here, and also we have the different statistics reported below that can be useful. You can evaluate the exposure, the hazard that has been calculated, and find out the main sources of uncertainty. And also you can obviously take a look at the risk that is presented through the risk characterization ratio. This value should be below 1 to have no risks. In this case, you can see this red line is telling you that 1 is here. So when the red line is in the green area, everything is fine. And you can see that it is quite far from the likely values of the risk characterization ratio. So this is a safe situation. I just show you a couple of things in the risk control part.
Please try to be quick, Alex.
Sorry?
Please try to be quick.
Yes, I'm just finishing. Just this, you can compare several assessment contexts and see how they perform in the different settings. And you can apply risk management measures that are derived from the SL database from TNO. So whenever TNO is including new risk management measures, these are automatically reflected in this list. And I stop here, but we could go on very long with this. So
I'm sure.
To Virginia, I guess.
Thank you, Virginia. Just a few minutes for Virginia.
Yes, I will do my best to be as quickly as possible. So good morning to everyone. Just let me share my screen. So can you see my screen?
Yes.
Okay, so I will briefly explain the main results of work package 5 case studies. So the first case study is on the occupational risk assessment of magnetite nanoparticles used as contrast agent in MRI application using the BIORIMA decision support system, as explained by Alex. So the overall methodology is here presented, and once the main physicochemical characteristics of this material, which is produced by Colorobia industry, are performed, then we perform the hazard assessment through the evaluation and the estimation of the DINEL using toxicological data of magnetite nanoparticles similar to the investigating nanoparticle. And then we identify contributing exposure scenarios for the exposure assessment through a questionnaire and interviews to workers potentially exposed to magnetite along the entire life cycle of this product. And we estimate exposure using the BIORIMA decision support system and the exposure models, and we can quantify risks. So for the ingestion exposure, no risks were observed for all the contributing exposure scenarios along the entire life cycle of magnetite case study. And the same was for the inhalation, where no risk were also observed for this route of exposure, while for the dermal exposure, no risks were observed except for the cleaning and the waste disposal activities performed during the use stage. And in this case, once we apply a specific risk management measure, in this case, nitrile gloves, no risks are observed also in this activity. For the second case studies, we performed an ecological risk assessment of silver nanoparticles using medical devices, also using the BIORIMA decision support system. And we selected catheters and wound dressings containing silver nanoparticles, and we estimate exposure in soil and water compartments along the entire life cycle of these medical devices. So for the exposure assessment, we use the probabilistic material flow models developed by EMPA, and we derive the predicted environmental concentration. While for the effect assessment, we use for the water compartment an ecotox database and the nano-species sensitivity weighted distribution model in the BIORIMA decision support system, and we derive the PNEC, the probabilistic PNEC. Then, using the software, the DSS, we integrate all these values and we estimate risk. And in our case study, and for the water compartment, no risk were assessed. So for the soil compartment, we use ecotox data for soil organisms, and after the application of assessment factors based on REACH guidelines, we estimate a deterministic value of PNEC, and we integrate with the predicted environmental concentration for the soil compartment, and we estimate the risk. And also in this case, no risk for soil compartments were assessed. So I would like to thank all the partners involved in this work and you for your attention.
Thank you. It was great, very clear. Even though the time is short, I think it was very clear, Virginia. Thank you. And Daniel and Alex. Any specific questions related to these results from the risk assessment work package? If not, we go to the next work package presentation, work package 6. So we have the outcomes from case studies from Carlos Fito. And after this presentation, we will have some time for a roundtable discussion and the wrap up of the project before we will start with the following sessions. So, okay, Carlos, you're ready.
Hello, good morning, everybody. Can you hear us? Hear me
yes,
properly?
yes.
Perfect.
You can see the presentation. You can start. Thank you.
Perfect. So now it's time for the work package 6. Work package 6 is about case studies to demonstrate the applicability and the impact of all the outcome from BIORIMA in the nanomedicine sector. So first, I just want to say that we have like 3 main points that I want to present today. Just a first overview of the activities that we have conducted until now in the framework of the work package 6 activities. Then we have also the results, so the lessons that we have from the case studies. So we have learned a lot from the companies. So there is a lot of things that the companies, the representatives along the value chain of the nanomedicine sector tell us during the life of the project. And of course, some conclusions about what we think about the future of the nanomedicine, considering the opinion of different companies and representative members of the value chain. So that's very important also in terms of the project, because we can try also to optimize some of the outcomes. So first is the project as such. Work Package 6 is about the demonstration of the performance of the outcomes. So we have a big impact considering the implementation of the risk management framework that we have developed in the life of the project, as our previous colleagues present. Then also we have different methodologies. We have a lot of tools, in silico applications that also we will have to transfer to the company. So there was a lot of things that we transferred to the companies and asked them about if they can implement in their real day-by-day activities. And then how we validate and demonstrate all these outcomes in the life of the project. So we select representative exposure scenarios during the life of the project. We study the implementation of the outcomes of the project in the companies that we have in the consortium. And of course, we go to the field, we go to different members of the nanomedicine community, ranging from companies to medical institutions, also research organizations, to see if they can implement all the activities that we have developed and all the tools that are the main outcomes of the project. And finally, we have developed a kind of guideline to support the companies and the nanomedicine sector to implement all the results with the support of the Work Package 6 members. So in summary, we perform like 5 main activities, so including the definition of performance indicators, then the validation studies in the companies. We use the validation to try to come up with some opinions and suggestions to improve the quality and the alignment with the requirements of the industry or all the outcomes. And then we develop this guideline to support the companies on the use of the tools. So our approach was to define key performance indicators, considering first a direct contact with the companies, and we tried to implement the outcomes considering key indicators, as for example, applicability, usability, scalability of all the results. And also we visit some of the companies to check, for example, if we can implement the tools to, for example, make a risk assessment or to conduct an exposure analysis without the need of going to the companies and measure using expensive devices. Then we did something similar in the stakeholders. So we went to different companies, we implement key performance indicators, and we organized different webinars where we asked different questions to representative members. Up to 40 members of the nanomedicine community were consulted, and we asked about if they can implement tools, if they are happy with the results of the project, if they think that they can use the tools in the future to support risk assessment or to perform, for example, an in silico analysis or a toxicology analysis. And all this information was compiled to analyze which are the opinions and lessons that we can learn from the different companies. So finally, also we define a kind of fact sheets about the different tools. It's a kind of summary of the applicability of the tools, the regulation that you can use the tool for which, and then also we implement a guideline that has been transferred to the companies in one of the deliverables of the project. All of these things was because we detect different barriers in the companies and in the nanomedicine sector. So the first activity that we performed was to try to identify which are these barriers. So we asked all the people and we analyze and we observe that there is a lack of information about different materials and synthesis approaches. Also, there was a lack of information to conduct toxicological analysis for the long term, for example, toxicology. There are a lot of tools at the moment, but there is a lack of tools to implement it to support risk assessment in the nanomedicine sector. And also, there is something important as the social acceptance and the consumer acceptance of all these tools. So we try to check which are the reasons of all these barriers, and we try to solve this barrier and overcome these barriers using the outcomes of the BIORIMA project. We know that the nanomedicine sector is a reality, so it's a big market. So we can see that it's more than $150 billion last year, and it's been increasing a lot so far. So we think that the outcomes of the project will be very important. So it's a reality, and all these outcomes can be implemented in the different companies that represent the nanomedicine sector and also all stakeholders. There are a lot of key players, but of course there are a lot of small companies that are using these kind of bio nanomaterials. So you can see that there are large companies, but they represent only 1% of the big nanomedicine sector. So we have small and medium enterprises, up to 30% of the companies. So these companies will be exposed to nanobiomaterials, and it means that the project will have an impact on all these projects. Then, concerning the materials, we observe that up to 45% of the products that they use are methyl oxides. And considering the type of products that they develop, of course, drug delivery was the start. So we have like 40% of the companies producing drug delivery products where you can use nanobiomaterials. So that's the first lesson that we understand from the company. The second was about the concerns. So one concern they have is about the risk assessment. So risk assessment is BIORIMA. So BIORIMA also, we anticipate in the past that will have an impact on all these projects. Considering the perception of the results of the project by the stakeholders and the companies, we saw that the companies in BIORIMA think up to 70% of them that is very important for conducting risk assessment and industrial facilities, but of course is also very important, have an impact on the regulatory implementation. So the BIORIMA have been a project that have a big impact on the companies that we have in the project in our first case study. Secondly, also we try to analyze if our tools are robust, and we analyze the performance, for example, of our tools to support the risk assessment. So we saw in the past that we have very big correlation between our estimation using the tools and the real measurements in the campaign. So these kind of tools can be used by the companies to support risk assessment in the workplace. So that's also very good news for the users of the tool. Concerning the framework, we saw also in the webinar that we did with the questionnaires and the online surveys that 100% of the attendees think that using the rich management framework will be very important to support, for example, rich implementations. And also they think in the majority that it will be not a problem, the implementation, because it's very easy to use by the different companies. That's also very important for us in terms of replicability and transferability of the project outcomes. Finally, also we have the decision support system, and we saw that much of the attendees also think that they can implement this support system to support the risk assessment, risk analysis, and at the end to support safer bionanomaterials in the workplace in the future. Controlling the exposure will be very important for them. They can use the support center for that. And also they think that they can analyze and implement risk mitigation measures as one of the key outcomes of the decision support system. That's also a key result within the life of the project. The overall vision that we have until this 4 years project is that there is a clear perception of the usefulness of the outcomes of the project by the companies and the nanomedicine sector. There is a high expectation and interest from the stakeholders on using the tools. And we thought, according with the opinion that they asked and they sent to our colleagues, that they will implement in the future all the tools to support risk assessment and risk management in the nanomedicine sector. So finally, we have up to 9 big results, tools, in silico models, in vitro testing, SUPs, and all of them will be implemented by the nanomedicine value chain. in somehow. And we think and we hope that in the future all these colleagues can benefit from the project just to reduce and mitigate the potential risk of this kind of materials. A big impact have been analysis from the companies. They can design safer products and materials. Also, we think that we can improve and refine the roadmap 2022 concerning the use of the nanomedicine and the nanobiomaterials in the medical sector. And of course, the project will support safer and cost-effective materials in the future that will arrive and achieve the industry. In summary, there is a guideline and tools that we can offer to the stakeholders from the nanomedicine sector. We have a comprehensive framework that can be implemented by the nanomedicine sector to reduce the potential risk of the nanobiomaterials, to support that nanobiomaterials reach the market and reach the applications in the medical sector. The BioRIMA facilitates and improves the performance of current applications to support risk assessment in the specific nanomedicine sector. And of course, we have a clear impact in the nanomedicine supply chain according with the results of the case studies. So thank you for your attention, and now we are open to all your questions.
Thank you, Carlos. So this was also very clear and in sharp time. Great, thank you very much. So with this presentation, we have closed the presentations of results from the BioRIMA project. I would like to thank the project monitor for being here, Professor Marcello Cacace, the advisory board members who are here, and the external stakeholders who belong to all our stakeholder groups for being here. We can open up the discussion on the results of BioRIMA. If you have any questions or comments, please take your word now. After this session, we will have a short lunch break. And then in the afternoon, we will have two sessions. The first one is about the results from COVID-19, let's say, link to BioRIMA. So the research within BioRIMA, as well as the research in nanosafety projects, can really give important knowledge, information, but also methods to real world needs. And for people to understand how close to us are these real world needs, we took the example of the COVID-19. And in the third session, we will have the nanosafety cluster community event, because we want this closure of BioRIMA to be something that opens up to further opportunities of course, cross-contamination among the different expertises, which are now represented by these 60 participants in this moment. So with this, I just would like to ask the stakeholders or Marcello or the advisory board members if you have any questions, and start the discussion of BioRIMA results. If Lang is also here, maybe he wants to say something.
I think you can hear me now.
Yes, Marcello, thank you.
No, I was, must confess, I was impressed by the fact that each work package performed, in my opinion, very well. So the picture which one can gather at the end of the project is really overarching and covering all the principal issues of nanomaterials risk and related problems. I think, I mean, this it should be emphasized, the fact that the results are there and they are available to the scientific community, because this is, in my opinion, is important. And as everybody could feel in wake of the COVID pandemic, the problem with fake news and non-scientific assessment of problems, they well may fit the problem of nanomaterials, as many of you may recall from the past debates on the safety of nanomaterials. So this is very important that the results and the framework that you provide is brought to the attention of all the interested stakeholders and government, to say, not least. So there is nothing which I can say that personally I am really satisfied with the project outcome. So this is I want to tell to everybody who is attending the meeting, because it's a sincere these are sincere congratulations to all of you. And as for the technical problems in fulfilling the deliverables and things like that, I think this can be discussed in the review meeting, which you will tell me when it will be held. And I suggest you to have it as soon as possible, so that you take advantage of the fact that you have all fresh memories of all these materials, and so we can discuss it more easily. So again, I am satisfied from the results that you have presented in all work packages. There are no at all, not even one work page which has revealed some problems. So again, I wish you to know that this is my opinion, and I will transfer this to the services, to European services. Thank you again.
Thank you, Marcello. This is very encouraging from your side, and thank you very much for these kind words. I would leave the word to some of the work package leaders if you want to say something to Marcello, or we can wait for.
First, a quick word from
you. Yes.
As coordinator. So it has been a pleasure to have you with us, Marcello, and it was a pleasure running BioRima in spite of the lockdown and everything. And as you can see, we have achieved an enormous amount of useful work, and beyond that, we have also contributed to the COVID-19 effort, and you'll hear about it later. So I hope everybody to stay on to see what we have done in terms of fighting COVID, in terms of assessing the safety of nanobiomaterials that are used in some of the COVID vaccines, and to contribute to an understanding to the after effect of COVID. In other words, the pathogenicity of long COVID. So those are the three areas that we have done with BioRima, and I think they are very timely, they are very useful. And as you mentioned about fake news, initially about COVID, we all think, and everyone in BioRima think about way of fighting COVID. But as it happened, as the problem unfolds, we also have the problem with those that are hesitant about the vaccine. And the main argument is about the safety of the material as well as the drug. And this is where I think nanosafety come in very nicely, because that's what we've been doing throughout all our projects. It's about the assessment of the risk of the material. And in this case, the BIORIMA, they are the very materials that are used in some of the vaccines. So more of it later. Anyone else like to have a few wise words now that we are closing the project?
Janeck, I just want to say to Marcello, I thank you also for your very kind ways all the time. And I think it's especially important with your words, because we know that you have such a large capacity. I mean, you have a very, very extensive knowledge in this area. So when you say something, we really listen, and you have always been a very kind and positive person. That's very nice. Thank you. Thank you.
Hi, Terry.
Oh, I'd like to say something, if I may.
Yes, Terry.
Yeah, Marcello and I are ancien combatants in the EU research going across many frameworks. And I looked at this project, and I think almost every work package not only achieved what it promised, but it's actually exceeded what it promised. And there were lovely examples yesterday, and I was rather hoping you might include that in your report, please.
Thanks. I think you may recall when the story started about studying the safety of nanomaterials. This was time passes very, very rapidly. So we are really close to, I believe, to the 15, 16 years ago. Time really has flown, and I mean, I'm really happy to be here to witness the progress made. I mean, this is really an achievement. I am happy because of that, really. And I'm happy that I had the opportunity to know most of you, basically all of you present in this project and this meeting. And really, I wish you all the best. And I hope we can continue somehow.
Thank you.
Thanks.
Before the next question, I would like just to ask everybody to stay for the group picture after we are done with the comments, and then everybody will turn on the lights and we'll take a group picture. But before that, is there any other comment, please?
If I may just add one short comment, I also want to thank you, Marcello, for your kind words and for staying with us. And I want to thank Lang, I mean, in particular for bringing us all together. I think it's important to recognize that this projects with 40 partner institutes, they don't sort of automatically fall into place. I think this there is a history, and we are all somehow connected to this history. And I mentioned in my talk the Marina project, which I think is sort of BIORIMA 1.0, and we are now sort of in BIORIMA 2.0. I think it takes time to learn how to collaborate and how to work in a multidisciplinary way. But so apart from all the results that we have achieved, I think one important accomplishment is that the fact that we are working together. And so, Lang, I hope you are ready for BIORIMA 3.0, because we are counting on you. And I think it would be a pity not to develop and to work with the momentum that we have now after more than a decade of working together, either specifically addressing the needs of the pandemic or in other ways addressing the issue of nanomaterials, biomaterials, advanced materials, and other materials on the horizon. But yeah, I think it has been a great experience to work with all of you. So thank you.
Thank you, Bernd. I think yes, this afternoon we will have a brainstorming. How can we move forward? And I think no matter how we try to be realistic, reality will always catch us where we least expected it, and COVID is one. issue. And it turned out that we do have a place and we do have a voice to say in a pandemic like this. And I think this is something that biomaterial, with our encouragement, the world has already used in nanobiomaterials. And I think that they are not too keen to talk loudly about this. And my point about this pandemic is that, as it turned out, we have all the tools to address it. In our small project, we have people who are doing exactly about it, about the assessing the virus potency, but also about the safety of the material. That wasn't what the commission envisaged originally. Initially, it was all about fighting the COVID, but now, as you see, it has taken another turn, and it's about safety of that thing. So there we are, and I think without saying any further, we should also accept the importance of dissemination and so on, because it really opened a very important case about dissemination. And I think we scientists, through the COVID pandemic, it has demonstrated that we have to address the public. We really have to address the public. We have to move away from our comfort zone of talking to our stakeholders, who are mainly scientists anyhow. So and the discussion among scientists are always following the rational framework. And you see now, we see it on TV screen. The general public is a challenge to engage and disseminate. And that is a lesson I think we have to learn, is that in the future, we have to think about addressing the people out there. And you see the criteria that they have for talking to them. And I think, again, with all our work package on dissemination, we should be able to do it. That is my view, is that one massive stakeholder has been neglected, and that is just the public. Okay. So on that, I think, Lisa, we should have a lunch break.
Yes, well, first of all, then I'll ask you, everybody, to turn on your camera, if you can, so we can take some group pictures, then we go out for lunch. Thank you.
Lisa, sorry, my camera doesn't work. Sorry.
Oh, that's too bad, Flavia, but your name is in the... So everybody's on, okay. Okay, we're taking pictures, hanging there. Smile. Even if you're hungry, smile anyway. Okay. And thank you so much. We'll come back at 12:30 for the session number 2 on COVID-19 related experiments and the response to real-world needs of BIORIMA. Okay, so see you back at 12:30. Thank you.
Thank you.
Thank you.
Thank you.
A dopo, buon appetito.
A dopo.
Bye, thank you.
Okay, so welcome back, everyone. It's time to kick off the next session, and I will chair the session together with Lang, who I think should chair the session in Italian, actually, since we are evidently in Torino, but I will do this in English. So we have 4 presentations. I will give the first short introduction, and then we have 3 additional presentations, as you have seen from the schedule, by Anna Costa, Terry Wilkins, and Janeck Scott-Fordsmand. And then we'll have time, I hope, for some further discussion on this topic, a topic that was not foreseen for obvious reasons when the project started. But despite the lockdown, we decided that it would be very appropriate to address these issues. And in fact, we think that nanosafety projects in general, there are several, as you all know, are very well positioned to address the safety of vaccines, of personal protective equipment, and so on. Not only with respect to this pandemic, I surely hope that it will be over soon, but also in preparedness for future pandemics. So in 2020, we established something that we call the COVID-19 task force, which I chaired together with Anna Costa. And it's a small group consisting of the work package leaders in BIORIMA and Professor Terry Wilkins from Leeds. And we had a couple of meetings. We issued a mission statement and so on. I will spare you the details, but the point is that the partners have taken this to heart, and there have been some very important activities in the project, and we will hear more about that, as I said, from the other speakers. Let me start with this figure. This is from an ACS Nano review published also in 2020 by Karsten Weiss and Marie Carrière and others. And it sort of nicely encapsulates the different areas where nanomaterials may come into play in personal protective equipment, possibly in face masks and so forth, but also in terms of surface coatings in the hospital setting or in other public spaces. And last but not least, of course, in the mRNA vaccines, which I will touch on very, very shortly. We discussed yesterday the issue of silver in face masks, but another rather contentious issue is the issue of graphene in face masks, and I just want to briefly touch on this. As you may know, Health Canada, the regulatory competent agent in Canada, decided in April last year to ban face mask containing graphene. In fact, it was one specific face mask, the one that is shown here from a company in Shanghai, which had been imported to Canada and widely distributed in schools and other settings. And Health Canada decided that this might pose a health risk, and so they banned the product. They were concerned that there might be release of graphene and it could lead to inhalation of toxic graphene. Now, interestingly, later in the same year, in July, the ban was lifted, and Health Canada issued a second statement saying that there was no potential health risk, and the risk of release of graphene was very low, and so on. And other graphene-containing face masks are now also permitted for sale in that market. This is just one example. I'm neither for or against this particular product. I think that vaccination is the way forward. Social distancing and face masks also have a role to play. But just as an example where we as scientists really should also make our voice heard. I mean, we have discussed iron oxide and other materials in BIORIMA, but let's not forget that actually graphene oxide, and with and without pegylation, was also one of the materials in the BIORIMA panel. So we do have some knowledge about these materials. This figure is also very informative. This is from a review by Kostas Kostarelos and co-authors published last year. And it basically summarizes all the vaccines that are currently being applied and which are based, as you know, on different principles. And some are inactivated or attenuated viruses. But in my own case, I have 2 shots of the mRNA vaccine listed there in the middle panel, and looking forward to my 3rd shot next week, actually. So these are lipid nanoparticles, and Terry will discuss this in much more detail in his presentation, but these are known entities. I mean, we have a great deal of knowledge about these materials and their different constituents. And in fact, if we look at the history of these vaccines, one interesting way of approaching this topic is to look at the history of the patents or the patent landscape. And this is a figure from Nature Biotechnology paper published recently. This graphic illustrates the patents that cover the mRNA vaccines, that is the lipid nanoparticle encapsulated mRNA vaccines. And you can recognize some of the major players here, and of course there is quite some history behind this. It's not that scientists all of a sudden came up with an mRNA vaccine. This has been a long time coming. Also from the same article, this is a figure showing the science underpinning the patents. So not necessarily all the scientific knowledge on lipid nanoparticles, but the science underpinning those patents that I just showed you. And it's interesting really to look at this figure in some detail. I studied this figure very carefully. I couldn't see a single mention of toxicity or safety. But if we look really closely, you can see there in the lower right-hand corner the word immunogenicity. So this is excuse me, this is in fact quite an important aspect. And again, it's a known aspect, that's something that we need to consider. That is the issue of potential immunogenic properties of lipid nanoparticles, the potential for complement activation, and so forth. I'd like to end with this figure. I showed this also in the session yesterday, but this is, I think, is very important. I mean, vaccines and all medical products, of course, have to go, are subject to the same rigorous safety and toxicity assessment. First, preclinical studies, usually rodents, as shown here, and then phase 1 studies in a limited number of human subjects, and so on, phase 2, phase 3, etc. This is from a very short summary in Science published in 2020. And again, I refer to Doug Green's editorial in Science Advances. And I think this is quite true when it comes to any drug, but in particular when it comes to a vaccine that will be issued not in the millions, but in billions of doses. We need to understand and have a good understanding not only of efficacy, but also of safety. So slow is fast and fast is slow. With that, I'd like to finish here and hopefully give some more time for the other colleagues in this session. So our next speaker will be Anna Costa from ISTEC CNR. Please, Anna.
Thank you very much, Bengt, for this very nice introduction. Do you hear me? Yes, so I'm going to share the desktop. Okay. Can you see the presentation in the presentation mode?
Not in the presentation mode, but maybe soon. Yes, now it looks fine. Thank you.
Okay, thank you. Okay, so good morning, everybody. Yes, I'm here for presenting my contribute, especially from Work Package 2, actually, to this BIORIMA task force. So as Bengt already introduced, this task force was born in 2020, so just after this undesired pandemic. And here is the mission statement that Bengt outlined since the beginning, and that was really addressed. So to identify science and technology solution across BIORIMA, which could be rapidly developed and deployed to reduce exposure and hazard posed by COVID. So with the same approach used for facing other nanobiomaterials. Of course, my contributor will be more focused on materials and on design alternatives that have been adopted for producing and testing antimicrobial nanophases and nano-enabled products. So nanosilver technology will be the main issue of this presentation. We know that this technology provides effective solutions in the human battle against microorganisms. It acts as a preventive measure that limits the contact with active pathogens and inhibit their activity. So the renewed interest in this material that is known since ancient times as colloidal silver is due to the dramatic consequences of this pandemic, but also to the more increased worrying for antimicrobial resistance drug. So which are the unmet needs for promoting these technologies, but also in general the nano-based technology? Well, of course, the need is of reliable and broad spectrum solution. In this case, broad spectrum antimicrobial measures that this dramatic need gave rise to many fraudulent, uncertified, ineffective, and often dangerous products in the market. So the nanotechnology have the capacity to offer safe and sustainable by design solutions. This is what we learn also in BIORIMA, both in terms of sustainability and safety. Of course, in terms of sustainability, the main important issues are what is highlighted in this slide. So the material should be low cost, should contain green ingredients. The technology should be easily scalable, easily applicable. The product should be stable. They should, of course, be produced with a high yield, including circularity in the strategy. In terms of safety, well, this is a specific need of this antimicrobial product. So they need to be selective as the drug need to be. So to increase as much as possible the antimicrobial activity without carrying on toxicity. So during the lockdown in 2020, we had the possibility in ISTEC to scale up a patented synthesis that match the criteria of safety and sustainability mentioned before. The silver nanoparticles are embedded in a matrix of cellulose derivate, a cationic quaternized hydroxyethyl cellulose polymers. Here is represented the structure that is considered a benign ingredient used as stabilizing gelling agent in cosmetic formulation. Thanks to this scale up, we could achieve in ISTEC a pre-pilot scale production up to 50 kilos per day with a live cost calculated of 0.06 euro per kilo. That is really very, very low compared to commercial product. We provide these materials in a suspended state, so as nanosol, with a mean diameter around 9 nanometers, but also as powder, as granulated powder by means of a spray freeze drying process. A joint effort between BIORIMA and us in a project led us to investigate the properties of silver egg, also when once integrated as coating of soft substrates, such as textile, and also thanks to electrospinning technique, electrospinning process, also as part of this woven non-woven textile substrate, where nanosilver is embedded, that are the materials produced by electrospinning. In BIORIMA, actually, the nanosilver technologies become a testbed platform for the BIORIMA task force. In Work Package 2, we introduced it as a Safe by Design case study, as explained before by Magda, comparing naked silver nanoparticles with silver egg. And this study was supported, of course, strongly supported by Work Package 4, with skin penetration and toxicological tests by means of open flow microperfusion applied to ex vivo and in vivo pig skin model, but also by ecotox tests using such non-target soil invertebrate standard model. But also they become, as you could heard before by Virginia, a case study for risk management framework in Work Package 5 that investigated the environmental risk associated with the disposal silver-coated face mask. Well, as Lang said yesterday, the show must go on, and so we introduce new nanosilver design alternatives in a new funded project called ASINA, that is coordinated by ISTEC. And now in a recent Italian national project funded by CNR, where also our task force leader Bengt is involved. So here is shown the systematic approach that we used in ASINA for identify the design strategy, and also of course, for identifying the best design strategy. So we made different comparison between different coating polymer composition. So we introduced new caping agent, new polymeric coating, and compare the reactivity, starting from of course, the physical chemical characterization and functional characterization. In this case, we compare the silver egg starting composition with a new polymer, curcumin, that is a proven antibacterial, antiviral phytocompound belonging to the flavonoid polyphenol class, that actually provide a silver sol with very, very low particle size, and as I will show later, with very, very high reactivity. But also we add to the silver egg and silver curcumin other polymers, in this case chitosan, and in the case of curcumin casein, in order to investigate some potential synergetic effects that can arise adding these polymers that are well known in also in antibacterial formulation for cream or cosmetics, something like this. We also successfully implemented the well-known design of experiment model to investigate some synthesis independent variables. In this case, varying the ratio between the sodium hydroxy catalyst and silver or egg concentration. Of course, as said before, we made a full characterization, TM size, crystalline size, hydrodynamic diameter, silver ion content, zeta potential, colloidal stability in different exposure media, and also, of course, characterize the functional properties. Functional properties I show here. We started with a general screening of antibacterial properties using as model Escherichia coli, gram-negative bacteria, and the gram-positive, I don't remember the name, Aarhus, a gram-positive bacteria. And we also tested the antibacterial properties of silver deposited on textile. You can see here the table. Difficult to comment all the data, but what is interesting to notice that thanks to this design of experiment approach, we found some alternatives to the starting GIEC, simply varying the ratio in the synthesis. And also we found that silver curcumin is very active as an antibacterial ingredient. And whilst comparing the silver egg deposited on textile with different treatment, so making abrasion cycles and also laundering cycle, we found that the antibacterial activity is almost preserved also after this treatment that of course mimics the use of this material. But looking to antiviral activity, thanks to an established collaboration with the University of Milan, we could measure the antiviral reactivity of silver nanosol and also of silver nanosol immobilized on textile, but also on hydrogel matrix against SARS-CoV-2. The SARS-CoV-2 was used was the SARS-CoV-2 B1 and the European variant before the Delta one that was isolated from nasal pharyngeal swab. And we could calculate the selectivity index that correspond to the compounds concentration required for having a product with an antiviral activity, but no toxicity. In particular, the selectivity index is the ratio between the cell viability by 50%, the CC50, divided by the concentration that gives the antiviral 50% inhibition. In this way, we could compare also nanosilver with a drug, chloroquine, that of course act with a totally different mechanism, but it was possible to compare such selectivity index. And that is reported in literature to be safe, to guarantee a safe use of the products if higher than 10. So we could test and see that nanosilver was in this case better, in this condition testing, better than chloroquine. We then compared also the other formulation with curcumin and casein, and we found a difference between antiviral and antibacterial, because in this case the silver egg seems again the better product, but if we follow the percentage of the reduction of percentage of replication. But in the case of curcumin and curcumin casein is interesting to notice that despite to their antibacterial reactivity seen before, they didn't show any cytotoxicity at the highest concentration tested. So the selectivity index can be higher than the value, because we don't have the value of cytotoxicity at concentration other than 300 micrograms for milliliter. So this, I'm sorry, this was only a brief and maybe not so systematic update of all the activity we made around this nanosilver. But the message that I want to give you is that we used all the expertise of BIORIMA for starting and progress of this case study, but also we need some further new efforts for the future that we ask to Lang to go ahead, because of course in principle the investigation of mechanism will be very, very important to optimize such a strategy. Thank you so much for your attention.
That's great. Thank you very much, Anna, for this very nice presentation and really showing how the research may continue even beyond the BIORIMA. I forgot to say this before, but of course, if there are questions from the audience, please use the chat to write out the questions if you like, because the idea is to take questions at the end of the session. So please make use of the chat, write your questions in the chat, and we will certainly address them at the end of the session. Thank you, Anna. Our next speaker is Professor Terry Wilkins from Leeds. Please, Terry, if you could share your screen.
Yes, signing on now. Let me find my presentation. Where is it? Where's it gone?
So for those of you who may not know this, Terry is one of the few, perhaps possibly the only person who has participated in every single framework program since FP1. So we are absolutely delighted to have you on our team, Terry, and we are looking forward to your presentation.
Difficulty in getting my presentation uploaded.
No worries, we can
wait
take
a minute. I can see the method.
We can take a minute and.
Yes, thanks to your presentation, I can take 1 or 2 slides out of mine, which might be a good thing to do. Now I'm going to share the screen. Get the presentation going. Share the screen. Away we go.
That's fine. Take your time.
Here it is.
Okay, here we go.
Here we go. Right. Fine, thank you. Right, if I get this side by side. I would like to say from the beginning that this is a co-presentation by myself and Marco Monopoli in the Royal College of Surgeons of Ireland. This has been a fabulous, short but very productive collaboration. I will talk about the partners in this collaboration when I get to the end, but I'll go straight to the start of the presentation. What the purpose of our particular part in the task force was to test BIORIMA's integrated risk management framework and our safe by design principles for understanding and reducing the hazards and risk of pseudo-anaphylaxis from mRNA vaccines using pegylated liposome nanocapsules. We wanted to examine if this new knowledge could be useful for application to other aspects of BIORIMA studies like other PEG products and PEG, PLGA and solid lipid nanoparticles in nanomedicine, and also to guide the future development of better nanocapsules and nanoparticles for medicine with regard to safety, stability and technology life cycle. Before we get too far in, we have to acknowledge these vaccines have been very, very successful and very, very important for saving lives. Some 8 billion people have been vaccinated with mRNA vaccines, and those are the two main candidates here. They're effective against all known variants observed so far. 2 or more doses have substantially reduced global morbidity and deaths. And I can say I've been doubly vaccinated with AstraZeneca and singly vaccinated with the Pfizer-BioNTech vaccine. The pegylated nanocapsules are used to deliver the vaccine, and these have been studied actively for pharmacological use for over 15 years. For example, used for doxil and sRNA treatment for cancer. More of that during my talk. The mRNA ingredient can easily be updated, and this means this vaccine can be adapted very quickly to new variants. Now, the risks so far, there are between 2.5 to 4.7 patients per million vaccinated have experienced life-threatening anaphylaxis, complement activation related pseudo-anaphylaxis. I will use the acronym CARPA as we go through. There may be longer term risks. There are other risks, the myocarditis and pericarditis, which I'm not going into in this presentation. And those affect younger people. It all started with the Wuhan outbreak, and it was an outbreak of a novel virus, fear that at the time that available treatments wouldn't work, and it transmitted rapidly in humans and it spread by travelers. Also, asymptomatic patients were successful viral carriers, and that early work was in January, Wuhan, but also the EU played a very big part in getting the genome of the vaccine, and as we now see, some of the earliest vaccines came from the EU. Right, so the purpose of vaccination is to train the immunological system before infection occurs. When you initial vaccination, you get a peak of antibody production, memory cells are created, and that's the primary response. And this slide doesn't mention it, but the initial antibodies are IgM and IgA. They're produced rapidly, and with time, of course, you develop immunoglobulins G. And the second immune response, the peak of the antibody production, and of course, the memory cell declines. I will not go into this slide because I think Bengt has already really had dealt with that. So the 4 main types of vaccines are those, and we're really going to concentrate on the Pfizer, BioNTech, and the Moderna vaccine. Okay, the core of the part of the mRNA vaccine is a lipid nanoparticle. You need an ionizable cationic lipid, cholesterol, a helper lipid to help form the structure, and a stabilizer to protect the particle. As we now know, this is polyethylene glycol. Now, the polyethylene glycol can exist in conformers like brushes or hunker down into mushroom shapes, and that's something that we looked at. So they're around about 100 nanometers in diameter. They protect the precious cargo, which is the mRNA, and the mRNA is released. The particles can be produced in quite a large scale, and I'll touch on that in a later slide. And of course, the cargo can be modified as and when required. So the pros are the world was able to get a rapid, fast solution. Scale up can be done at a high rate, as batch or continuous flow. The active ingredient, mRNA, is easier to scale up than conventional virus vaccine components. It's a new generation technology. I guess it's early days were in the 1960s, but it's become much more actively studied and put to use since the last 15 years. The cons has not been used before. It's mainly been studied primarily for anti-cancer treatment, and it's unknown to the public. Now, the structure of our review is here. We've got the motivation, the target audience, the methodology, particularly review of the scientific literature, review of BIORIMA data, and gap analysis. We didn't do any experimental work. We relied on knowledge and our brains to assess and collect the data from multiple sources. The specific analysis of mRNA vaccine and drug carpet studies were directed towards polyethylene glycol, its chemistry and its antigenicity, the pegylated liposome nanocapsules themselves, the the materials of manufacturing using safe by design analysis. And then we go through to recommendations and conclusions. So the mechanism of CARPA, take that out. I can see a corner of my slide better. So those of you who are familiar with normal allergy, the allergic, the anaphylaxis due to allergens involves mast cells, stimulation of mast cells, immunoglobulin E, and the allergen results in degranulation and all sorts of horrible things happening fast. As I know personally, in 2007, I suffered this as I discovered the hard way I'm allergic to lupine beans. And I have to say it was an Italian gluten-free pasta that did it. So I spent 24 hours in hospital, near to death, but fortunately adrenaline and the usual care processes kicked in. Now, the non-allergic, or if you like, pseudo-allergic mediated hypersensitivity, again involves the mast cells. The engineered nanoparticles interact with complement and cause a rapid cascade, C3a, C4a, T5a, degranulation, and something that looks exactly the same clinically as normal severe allergies. The difference is this one, you're over in about 24 hours, you're feeling tired, and you recover quite quickly. Here, the patients have to be monitored for at least a month because they can relapse. And so that's a description of the involvement of the anti-PEG IgM antibodies and how the complement activation goes, and the liver tries to cope with it. So what we did was, if you like, a big thought experiment using reverse engineering processes. The scientific literature review was done largely by the Royal College of Surgeons in Ireland. The patent and commercial and regulatory and literature review was done by these, but we've also brought into that our experience of designing and manufacturing siRNA drugs for cancer, GI tract cancer therapy. There's a project from 2009 and 2010 in the University of Leeds. So we did a detailed analysis of all the biological interactions. Model of product and process workflow was generated by Leeds, and we've applied safe by design in the integrated risk management framework criteria, which you can read in the excellent paper, which was led by Danielle. It's all there, and those methods are the ones that we use. We came to some conclusions. We've written a draft journal paper and a BIORIMA report. And again, that's a little summary of the CARPA process down there. The process for making these nanocapsules is actually quite easy. You take all the components, including the mRNA, and you mix them all up together, and they self-assemble to produce that, which is quite convenient. And then you have to separate these particles from everything else in the initial reaction mixture and filter them down to the final product size to make them uniform. So that's a very useful process. Now, I want to just touch base on our work in 2009 with King's College London. That's again our friendly nanoparticle, but in this case, we had an additional layer. We had targeting ligands. These were folate receptors to tack onto cancers, GI tract cancers, and we also had a lipid with an indium with a chelating agent to take indium-3 as an MRI imaging contrast staging. So we had siRNA anti-cancer drug there, and we used MRI-guided focused ultrasound to trigger the release of the drug. There's some examples of showing the indium imaging agent in the tumors. That actually got that technique here with the targeting linkers took it into the tumors in about 2 hours. You got more drug collected that way than by the EPR method. So we do have some actual experimental expertise on the manufacturing. This was necessary because companies keep the manufacturing process very, very secret. You can patent it if you like them, but then you teach all your competitors how to make it and get around your patents. So that's why those things were not readily available to us. So our conclusions are this. The product design and manufacturing processes of the 2 leading vaccines leads to well-optimized and uniform 100 nanometer products. PEG 2000, which has a molecular weight of 2,800 used, is not a natural product, has a regular OCH2 twice O structure. So that's it, that's the unit of the polymer structure. And exposure to the immune system can generate IgM and IgG antibodies. CARPA involves the binding of IgM to the liposomes, leading to complement activation. Subjects we've discovered through this database from the PharmaKB organization has it that subjects with glucose 6-phosphate dehydrogenase gene deficiency, G6PD, are at severe risk to PEG-induced CARPA. We would like to discuss some of these implications with our clinical immunologist friends, particularly Bengt, to assess if further work is needed. The repeated use of these vaccines long-term should be studied to assess if A, the incidence of CARPA is likely to increase. It might not, given it relies on a particular deficiency, gene deficiency, and B, do pegylated vaccines or drugs become deactivated when the world is repeatedly vaccinated with COVID-19 mRNA vaccines? The PEG, we've not assessed PEG PGLA nanoparticles yet. PGA is a copolymer of glucolic acid and lactic acid. So it produces a long chain that has an acid group at the end. This is biodegradable. PEG isn't. So there's some interesting questions to examine there. So in conclusions, BioRima's safe by design methods could be used to design better vaccine nanocapsules. I don't know if Marco's in the session or not, but I know he's going on with at least one project and hopefully another one, which could produce a better capsule not using PEG. So that's a secret, and that's some hope for the future. This is the first page of our draft publication, and I will leave it there. But my grateful thanks are to the Royal College of Surgeons in Ireland for stimulating and supporting this work. These are 2 students, Esperanza Gonzalez. Her initial review gave us a racing start into our study here, and Pearl Lancaster joined in to help. Both students, now they're graduated and gone on to be researchers in their own right. And of course, Marco for his outstanding work here. I'd like to thank you all for listening to our presentation.
Thank you very much, Terry. This was really an excellent overview, and I think a really important piece of work that the BioRima project may contribute. So as you say, we are working still on the publication and the report, but this information I think is very, very valuable. I'm sure there are questions and comments. Again, I just like to ask everyone to make use of the chat, and we'll have an opportunity very shortly to entertain those questions. I think at this point, we'll wrap up with the final talk of this session before the questions and this. So I ask Janeck, please, to share your screen and give us some environmental perspectives on COVID based on your knowledge and your experience, please. I don't know, Terry, if you need to stop sharing first.
Yes, I'm going to do that. Then switch myself off.
I think you need to stop sharing the whole screen as such. Probably, yes. I think that's right. Quite. That's
I think you see my screen now.
Yes, we see.
You see a presentation.
We see the slides. Yes, thank you.
Perfect. I'm going to make them into presentation mode here. Thank you very much for organizing this session, Bengt, and for organizing this all through the project, or at least when the pandemic started or panic started, whatever started, right? I'm very happy for this, and I thought it was a very nice opportunity to do something that we could all help society with in a very direct way. So what we did was that, I'm going to show only 4 or 5 slides, but what we did was we went back to our drawing tables now that we were confined to our home and getting frustrated walking around the table. And we thought, what can we do? What can we do? And we went back to our knowledge and thought about what we had in our bag here. And then one of the first things is that, you know, viruses, I mean, I'm going to say some trivial things that you will know all, but viruses, of course, not solely related to humans or mammals for that matter. They are, in fact, all over. So there must be knowledge out there to you. And in fact, when you think of immunology, it was actually based on invertebrates from the start. I mean, the first discovery was in fact invertebrates led to a Nobel Prize in that. So there's a lot of this, and invertebrates, of course, had an innate immune system that is very similar, if not fully similar to the human health, and has developed into that. And if you think of invertebrates, they have led to discoveries in almost every area of biology and medicine. I mean, all the way. And if you think of all the foundation developments, they are often discovered in a small worm or in a fruit fly or whatever, all the basic things. And they, of course, have not related to COVID, but of course they have the advantage that there's no placebo effect here. You have a directly biological effect. You can execute them rapidly. You can see what it is. Then, of course, you have the problem of extrapolating from this to that. But there's a lot of basic functions that are the same. And this is actually, there's also some differences that we also see now. But this is actually what led us to think, well, we can do something here, hopefully. So just to make it upfront now, so we produced 3 publications on this, and we made a special issue, and that's the reason why I'm bringing it up here. We have a special issue in Impact Factor 5, whatever, on invertebrates as models to human pandemics, facts and opinions. And what we encourage people to here is to really write. I mean, many places you have to produce data for papers, but here we are really interested in novel ideas that has some sort of logic or at least daredeviling in it, and just submit some nice papers there, you know, fantastic ideas, whatever. I don't think, maybe our idea is not fantastic, but this is what we want. We want to brainstorm to society and how invertebrates can help, not only this pandemic, but also the next pandemic, facts and opinions, and that's the way forward, we think. So I'm going to do the next. So the first thing we did was we went back to some of our nanomaterial studies and nanobiomaterial studies. And one thing we have realized along is that our worms, if you look down in the left corner, you can see a worm cell, and there's a nanoparticle in gray and some slime at the top in orange, whatever color it is on your screen. But that is actually a cell producing a cover, a coating of a particle coming into the worm. And the first thing we think, oh, this is, you know, protein coronas on cells, and it's interesting, and yes, they can see it and so on. But then we thought, and we actually noticed that when it was produced, it gave a feedback to the cell. So it covered the cell, the nanoparticle, it feedback to the cell saying, oh, there's no danger anymore, or a little danger, right? So it reduces the covered protein production for the coverage, And then it was like a cycle. But then we thought, I mean, how do they know nanomaterials? I mean, okay, they're exposed to sand and all sorts of dust, but what is it actually this? And of course, we relatively easily identified this protein. It's called leucinin. And from there, we spoke, what is it going to use leucinin for? Is it only for nanomaterials entry? Hardly, right? That's hardly the sole mechanism of this. And in fact, it shows that this is a pore-forming protein that attacks incoming particles, usually viruses or bacteria or whatever it is like that. And then we thought, what is the mechanism behind when an earthworm or a worm is exposed to a virus? And in fact, what it does is uses this protein to attack the membrane and make a hole into the membrane, and the virus is destroyed from there. So and it's been shown for many other viruses, cold viruses, and, other viruses, it's been shown that it works. So that would be a very nice mechanism. And then you can ask you, why doesn't it happen with the worms itself? Well, the worms itself, they had to adapt to this situation also. So actually they have a different membrane than we have. So you cannot use this protein directly in humans, because then we're going to kill each other or ourselves, right, if we take this protein. But it turns out that there's, I mean, some people, bright people before us has been working on this, and they actually made a non-toxic versions of this. So what we speculate in this paper is that you can use the non-toxic version, which has a very specific binding to some fatty acids in the membrane, and then we can attach something to there to make it a target. That, of course, acknowledge that there's a difference between the membrane of the human cell and the virus. And although the virus hijacks the human system for producing a membrane, it seems that the membrane of the virus is different. So it's not the exact same composition. So that is the thing we are going after here, that it's actually a different composition of the membrane. And you know, all of this is outside the spike protein. We're not talking about spike protein. We're talking about this basic of this virus, which is, you know, there's toxic and non-toxic versions of this. So that's how it came out that we did this. And we produced a paper on this, and we wanted to get it out for inspiration for people to try to work on with this, and we work on with this and so on. I think please just take it as an inspiration to this or new ideas that could be used. The second thing we did is that we also know something about invertebrates in general, and we sequence our genomes and so on. And then we start, you know, there must be something in the genomes that are similar, because this not only is you know, the immune system very similar, but of course the genome systems also have a lot of similarities. And of course, in the genome system, we also found the ACE promoter or the ACE gene in there, or similarity to the ACE genes. And we thought, okay, this is really interesting. So this is also a precursor for the spike protein, as you know. And so what can we do here? So we published it and we found it, and then we went on to study inside the project what is actually activating of these nanobiomaterials that we have worked with and other materials, what is actually activating and deactivating this pathway, angiotensin pathway. And then we studied a number of different chemicals, or nanobiomaterials, and one of them particularly stood out after these studies. And as a basic for that, we went to see the particles. You look at the right-hand corner, down in the right-hand corner, we looked to study all of these materials' surface characteristics to see whether we can find something to the activation, deactivation of this ACE gene expression. We haven't found the exact reason yet, but we are certainly on the target to this, trying to find what the activation is. We're not certain whether it would help something, but it's certainly a surprising results for us. And you can see, if you look at the right-hand top corner, we've tried to make an illustration of how this may be happening, what may be going on, and that is coming out. So that's it. And I think just a summary, because we need to keep time short, I know. So invertebrates have mechanisms to handle virus, not surprising, but there are something that can be helped. They have the ACE activation mechanism, and some nanobiomaterials cause activation of the ACE and deactivation of ACE. And finally, it's not the end of pandemic, and we should continue studying invertebrates to use to help human biological responses to pandemics and so on. That's it. Thank you, Bengt. Hope we was in time.
That's great, Janeck. Actually, we are perfectly on time, and
thank you.
I guess both you and Charles Darwin have a strong affinity for the earthworm.
Exactly. I'll try to stop scaring my machine.
I would like to give the word to my co-chair now at this time. Lang, if you would like to lead the roundtable discussion. Are you?
Thank you. And I think we had a very good few presentation that summarizes our commitment to the COVID effort there. And I am very impressed with Janeck and his take from the ecotoxicology angle. I think that is very interesting, very innovative, I must say. I never thought that you can actually find an angle from that. But let's see how people would respond to our effort in a round table. So I open the discussion now to everybody.
We may also certainly invite Marco Monopoli. You are present, I believe.
Marco?
Indeed, yes.
Yes. So if there's anything you want to add, then please do so. I'm also wondering, Miguel, if you are present. I'm not quite sure, but I'm sure you have some important insights also when it comes to surface coatings and inactivation of the pathogens. So these presentations that we just heard were just snapshots, but there is more knowledge in the consortium. So please.
So thank you very much for giving me the opportunity just to say a few words. Probably like all of us, we got really excited when the RNA vaccines were coming up. But obviously with our experience with the nanosafety, part of us was also wondering if there would be any adverse effect, but probably all of us were quite optimistic just because with all of this information that we gathered in the nanosafety cluster and all the other European projects, I'm pretty sure that all of us were quite comfortable. Regarding the PEG, I'm the coordinator of a European project called NanoCarb, and the PEG is often used in nanomedicine just because it's available. You can get a different size, different functional group, and that's quite heterogeneous, and it allows to be tailored to a wide range of nanoparticles. However, we do know that the PEG is not bulletproof for corona formation, and also we can see that this very, very rare interaction with the blood proteins can lead in some rare effect that could lead to anaphylactic response. So I think that nanomedicine, we should be very proud. Some of us work in the field of nanomedicine, some are in nanosafety, other in environmental nanotoxicology modeling. But because of the knowledge we gained so far, all of us contributed to the EMA and FDA approval and confidence of this vaccine. But at the end of the day, we just need to, as Bengt said, we just need to think about the next stage. How can we make these safer? Can we use more biocompatible polymer, or can we put a cap on this polymer? For example, in nanocarb, we use monosaccharide or glycans that will increase even more the biocompatibility, and that's what I think it's where we should go. Where especially now that we're going with the 1 dose, 2 dose, 3 dose, and so on. So that's, I think it should be the eye and the angle we should take for future projects. Thank you.
Yes, thank you, Marco, for this important additional statement. Maybe we should coat the particles with earthworm proteins. I'm not sure, but some alternative to PEG should be explored probably. Miguel, I know that you and also our colleagues in Hong Kong, I don't know if they are present, but you certainly have also extensive knowledge in this domain. So is there something you would like to add?
Okay, our approach has been basically in a manner that will minimize the exposure to the people, and it is targeting to inactivate the virus. So the idea is to use filters to circulate the air in rooms, chambers, or in air conditioning circuits, so that when the virus is passing through that filter, it is not absorbed, because it is not an absorption filter, it is a catalytic filter. So the idea is that by oxidative stress damage, you affect the outermost layer of the coronavirus, and then renders it unable to bind to the cell and to load its virus inside the cells. So this is the idea. In the case of Hong Kong, the group of Tin Yeong, he's working on photocatalytic activations. In our case, we are working on thermal catalysis activation. So the idea is to do to generate this oxidative damage on the virus. You don't really want to burn it away, but once that you render it unable to infect, that's good. And then the balance is that people will be constantly generating virus, and these filters will be constantly inactivating the virus.
That's very interesting. And this would not be necessarily only, I mean, this would be regardless of the variant of the virus, obviously. It's a more universal approach.
Yeah, indeed. This is also one of the attractive aspects of it. And in general, I mean, there is a general value on a general biological quality of air.
Right. Yes. Thank you. Anna, you had a question, which is it something you would like to who is it addressed to?
No, it is a thank you, Bengt. No, it is addressed to all the partners. It is just a curiosity, but that can become maybe interesting for some of you, because the researchers, the professors that makes such antiviral test with SARS-CoV-2 has access to all the variants, and so I asked her only for curiosity if she noticed a difference in vitro test, in vitro model she tested, and she said that she noticed a lot of difference between the variants Delta, Omicron, and the first one I mentioned before, Beta. So I don't know. I'm absolutely not equipped for hypothesizing some reason, and so I was wondering if someone could have an explanation, or if this can be an issue interesting to investigate at some level.
Is this? Are you talking about differences in susceptibility to the silver nanoparticles?
No, not to the silver nanoparticles.
Oh, okay.
She was referring, no, for our test she used only the beta variant. She was referring to some other kind of test, I suppose. She only told me serological, antibody, and pharmacological. So I don't know which kind of in vitro, actually in vitro model she tested, but she, I know that she tried to stimulate the immune system with some model. I don't know exactly how.
Well, these variants have mutations in the spike protein, so they vary in terms of of the spike protein and how well it binds to the different receptors. Of course, we now know that ACE2 is the main receptor, but there are other cellular receptors. But would someone else like to comment on this?
I would like to comment with this.
Sure.
I think especially with maybe, I mean, this is out of my field. I'm just speculating on what Anna said. Maybe the last variant, the Omicron variant, has so many mutations that it also have a change in the corona itself. It's not only in the spike protein, which also have implications for what we do with it. And I mean, I think no matter, I think, first of all, what I think, sorry, just jumping back a bit, what I hear from Miguel and from Anna and from Marco and all this, I think it's very nice that they often refer to the idea. They had an idea, they pursued it. And this is what we really can do in this field here of nanotechnology, that we really know so much about these small particles, be it virus or nanomaterials, that we can come up with good ideas to pursue this. So the second thing is, of course, not only spike protein will, probably no matter what we find, unless we kill them all, like with Miguel's method of kill them all, they will mutate and mutate and mutate, also in the corona and so on. We need to find back to something that more fundamental with virus compared to bacteria, compared to human cells, and that's probably where we need to target. And apart from not being biologists, I think, or being biologists, that's what we can really do something with. Sorry, a very general statement, but.
Thanks, Janeck. I'm not sure if I can, well, if I see any more questions at the moment. We had one question earlier from Terry, but I think you addressed that. Lang, please, maybe you could take.
Janeck, you are right. I think, as you can see, the speed of mutation of the virus is very fast. And there's no way, if we are going down the current approach, we can mutate our vaccine as fast as the virus. So we had to think, we had to take that as one of the fundamental risk factor, is that if we're going to design a vaccine or any treatment against the virus, it should somehow be so fundamental that it can knock out the virus in various mutations. Because otherwise the virus will always be in front of us and we're playing catch up forever. And I think that is, I think, a very important message that we have to convey to the community, that we should come up with treatments that somehow anticipate the speed of the mutation, so that we can address all the mutation if we can.
Ben, can I come in there?
Please.
If the speed of the mutation is important, the important thing is the speed of creating a new mRNA vaccine. The speed of that is potentially very good. And if we could find a better, more general nanoparticle, we got it made. And I do believe the approach that Marco is going with glycosylation, learn from nature. Proteins are glycosylated to stop the body rejecting them. And so we could use the normal glycosylated protein model as a model for developing a better nanoparticle or nanocapsule for carrying the vaccine. So fast vaccine, better particle, I think, is a way forward.
Yeah,
thank you, Terry. Yes, yes.
Yes, no, because I mean, this point is very important. So there is indeed a way is basically running with nature, fighting all the mutations. But the other thing is to minimize the chances for mutation. And in that sense, I believe that along with the vaccine strategy, this strategy of inactivating the virus is very good, in the sense that the less people get infected, the less chances it has to mutate, because there will be a lower population of active virus. So when you render it inactive, and that really, it is in the area of the risk, you are minimizing the exposure. Or well, I mean, you are exposed, but it's no longer relevant because it's not active.
Okay.
And that
will two-pronged strategy then is a good way.
So this combine is
you do both. Thank you.
This is important. Actually, probably most of you don't know this, but before Nanosafety and before all that, I actually started out as a PhD student doing HIV research, and of course in the early 1990s, and we were always one step behind, and I think HIV, of course, is obviously even more prone to mutate, and has been a long struggle. But I think I'm also quite intrigued and inspired by Janeck's presentation, because we are not, as you said, we are not the only hosts that are infected by viruses, and so there may be even more fundamental defense mechanisms that immunologists and virologists overlook, because the favorite model is always the mouse, and maybe the primates and then the humans, of course, but there may be more primitive defense mechanisms that we should also consider and learn from. And also to keep in mind that these vaccines will not only be used to fight virus infections. It's very likely that we will see anti-cancer vaccines based on the same principles. Now that we have a billion people who have been used as test subjects for these materials.
There's a question there, Bernd, from somebody in the Thanos.
Yes, hello from NTUA.
Yes, please go ahead.
Yes, regarding the presentations of antiviral properties of nanosilver, regarding the yesterday's presentations and today's, at least I haven't understood correctly about the actual mechanism, whether it's attributed to the nanoparticle itself, as in physical nanoparticle, or to the produced ions, as in certain cases of microorganisms and bacteria, or regarding this.
Yes, that's a great question. Maybe Anna, you want to address it. I can start with a few words. We actually think, based on our experimental findings, we are currently revising a manuscript. We hope to get it published soon, but according to our findings, it is a silver nanoparticle effect and not a silver ion effect. In terms of, and we think that the mechanism is that there is a strong affinity. Of course, silver binds to DNA, to RNA, to proteins. I mean, this is widely exploited in cell biology, but we think that there is a case to be made for the denaturation of the spike protein when it is in contact with the surface of the silver particles. So we are favoring that mechanism, and we don't think it's the silver ions because we don't see denaturation of the spike protein with silver ions.
I tend to agree it wouldn't be the silver ions, and I'm waiting for Miguel to come in on this. The silver metal has a lower oxidation state, and it's probably that reducing characteristics of the silver particle that's doing the deactivation. And it will be dead easy to test with experimentally and actually with some of the nano Q-cell work we did. Incidentally, we have experience of putting it into hydroxyapatite coatings on nanoparticles, coatings on hip joint shafts to protect against nosocomial infections in the first few weeks of use in vivo. What we discovered is it worked for a while, but it slowly, the silver slowly converted metal, slowly converted to silver ions and became part of the hydroxyapatite structure.
Yes, I was wondering, Anna, the silver particles with the cellulose shell, do they easily dissolve and release silver ions or not?
No, actually, no. Yes, this is a good question. I didn't mention in my presentation because my only evidence are indirect evidence about the possible mechanism, and we try to confirm what is reported in literature, which are the most recognized hypotheses in literature. So in case of antibacterial effect, is predominant the silver ion contribute, whilst in case of antiviral effect, it seems a particle particle-based contribute due to the capability of silver in this case to make a film around virus, blocking the interaction with the binding receptor. So this was confirmed by our experiment only in indirect way, whilst Bengt in the paper showed in the work show yesterday by Govind, maybe was almost more validated. because when we mixed the silver with when we put in contact silver with infected cells, so cells already infected by virus, we didn't note any effect. Whilst when we pre-treated the virus with silver and then we put in contact with cells, we achieved the expected block of inhibition or block of replication. Right. So it is a confirmation of the mechanism reported in the literature that nanosilver block the point of entry of virus. But I remember a very interesting test that shown yesterday, or that Bengt showed me, that in case of other particles that have the same capabilities to film the virus, they didn't notice any reactivity. So for sure, one mechanism is the capability of such nanoparticles, very small nanoparticles, to make such uniform film around virus, but not all nanoparticles can do it, because maybe I don't know if it is a chemical absorption or physical absorption, or which kind of prevail. It depends on surface charge or not, but for sure the silver is very good in this. And also our silver, to respond to the question, our silver didn't release any significant amount of ions. We achieve almost a 99% of yield during the conversion of silver into nanoparticles. And when we obtained the suspension, and when also we disperse in the media, we didn't notice any increase of the amount of ions.
Okay.
Yes.
Right. Thank you.
I had just a point.
Yeah, okay. We are close to running out of time, but Magda, a short comment, and then Panos, if you had a follow-up.
Yes, about the mechanism, the interaction with the protein corona, with the protein, the spike protein. Sorry, it appears as fundamental because we also tested our silver nanoparticles against not enveloped viruses, and it doesn't work with not enveloped viruses. So we tested the polyoma viruses, and we had no effect against this kind of viruses.
Thank you, Magda. Panos, did you have your hand is raised? Did you have a follow-up?
Yes. It's not more of a question, but more of a comment. If I understand correctly, we have it's more like an inhibition than an inactivation of the virus or killing of the virus, if I understand correctly. So could we say that there is a side dependency in the silver nanoparticles? And if yes, if this could be a good research in terms of formulation, of studying which size distribution of silver nanoparticles are effective in inhibiting virus and which kind of virus. Just a minor comment.
Yeah, thanks. That's I think that's a good point. In our studies, we have evaluated silver particles of different sizes and silver particles of the same size with different surface coatings, and we hope to publish this soon. But yes, I think it's an inhibition of the infectivity, because it's we think that we are essentially destroying the spike protein, and therefore the virus cannot infect the host cells. I think we can go on at length, but since we have another important part of this afternoon session, I think we should probably close here. But thank you everyone for participating and in the discussion. Is that okay, Lang? We may turn over to you and Andreas probably.
Thank you. I have the last word to say. Thank you, everybody. And I think the message I would like the Commission and through Marcello is that we clearly demonstrate that we have what it takes to have our voice heard in this vaccination and virus strain. Clearly, the expertise through Anna, yourself, Marco, and so on are very clearly evident that we have what it takes, and we strongly believe that we can contribute to the debate about this. And this is what I want to say. And with that, I thank everybody, and I thank the Commission for asking us, and the enthusiasm of everybody in joining in the fight against the virus. Okay, with that, we would turn this to Andrea. Andreas, is he about? Yeah, okay.
Thank you very much, Lang. I would say you already made a perfect introduction with this session. So it's great to see that there is really impact on the one hand delivered by the project, and on the other hand, a lot of work in front of us where we really want to brainstorm on what are the key expertise we should have at hand. Also to make it an interactive session. Susanne, you still are in the presentation preparation mode. Maybe you can shift to the other one. Perfect, thank you very much. We said we want to make this session really active for all of you. We know that now there are nearly 60 people in the room, which means if for you, Lang, it would be okay that we take the nice discussion we just had as the already the introduction for the session. We will make it kind of interview between, so to come forward with the session. Would that be okay for you, Lang?
Yes, yes, definitely. Go ahead.
Perfect. Okay, so Susanne, you might switch. Yes, thank you. The interactive session, we have 4 points we want to go through. And for each, you will get some time. The first one is a little bit to get you also familiar with this virtual whiteboard. It's called Mural. Susanne will introduce it to you shortly, and then you have the who is who question. So just prepare your name and your affiliation that you can put there. Collaboration offer. Okay, what is my expertise? Maybe 1, 2, 3 words, not more, about your expertise. We want to get a kind of real overview what the things in terms of doing nanosafety related work in this section with nanobiomaterials you would put on the plate to use for the future collaboration to tackle challenges like the COVID pandemic. Collaboration topics, please think really visionary, not only what you are working on now, but also what you promised or you envisage to work on in the future. And of course, we have one last chapter in that about the public communication on the safety of nanobiomaterials. On the one hand, to identify even some communication challenges, and also to make it possible that you can volunteer for some activity, either throughout the nanosafety cluster or in any other way of collaboration. This is the short introduction from my side. Susanne, I wanted to hand over to you, if okay.
Sure, that's totally fine. Thank you very much. So this session is really not to cut off the previous discussion, but to continue it and just make use of an online whiteboard to collect also feedback and inputs from all participants that are now in the call. So these 4 topics that Andreas just described will be shown on this mural whiteboard. So you see these 4 blocks that we will work through, and I will guide you once you're in the mural and also tell you how much time you have for each activity.
Susanne, I just put the link into the chat.
That was a bit early, so please still follow my explanation before you go to mural, because I will show you how it works. So it's quite easy, hopefully, for all of you. So you can zoom in and out just by scrolling with your mouse. You can move around the whole whiteboard via drag and drop. And to add a sticky note, you just need to double click and then it will appear, and then you can start immediately typing your comment, feedback, whatsoever. And we just kindly ask you to add a new sticky note for each new comment so that it's still readable and not too small in the end. So yes, this is the slide with the link. I kindly ask you to stay logged in here also in the Zoom, because we will use Zoom for the audio connection. And yes, you should be ready now to enter Mural and click on the link provided by Andreas. You don't need to register, you can just enter as a visitor. So I hope it already works for most of you.
Can I say a few words to encourage people to join this? Because I think this is a very important thing, because the outcome of this is going to be fed into the Nanosafety cluster, and it will influence future calls. And yes. Recently, the Commission is very keen to listen to the voice of the Nanosafety cluster. So I think there is something in here that we can help to shape and formulate the future course. So please do participate and do help, and I'm sure this is very useful.
Yes, thank you, Lang. That's a very important comment. So if everybody is fine with the link, I will stop sharing my slides so that I can also switch to Mural. And I think we can start with this very first activity that is really just meant to make you familiar with this tool. So the first one is about who is who. So I kindly invite everyone to add a sticky note with your name and organization that you are representing. And I will give you 3 minutes for that. That should be doable for all of you, I hope. So if you experience any difficulties by using this online whiteboard, just let me know, and or type a question in the chat in Zoom. That's also possible.
Thank you very much, Susanne. It looks really very, very busy already in the Mural. I like this interactive part of such meetings. I hope that everyone is also happy with this kind. As Lang already mentioned, after the who is who, we want to identify specific expertise that is needed along. And I think, Lang, it would be a perfect timing if you would give a short impression what, from your experience now, from BIORIMA exercise, you would say would be the greatest things that for such collaboration could be mentioned there. So just to give a little bit the starter for the people here in the room. Lang, are you still there?
I'm here, but shall I talk? Shall I say now from my own perspective, or do I have to write through the Mural? And
no, you can talk, Lang. It's fine.
Oh, well, my personal perspective as the coordinator is it has been a pleasure working with all of you. I think that is the first thing. As you know, that BIORIMA is not the first project. We worked with each other before in Marina. Okay, that was also a very interesting and very successful project. And in a way, most of the core people in BIORIMA are from Marina. So that is basically where I would come from. On the intellectual and scientific level, I think the nice things about BIORIMA is that everybody is prepared. Everybody knows exactly what they have to do, and they enjoy doing it. That's the most important thing. And to me, it was a pleasure coordinating this project, because there's never any massive issue. People just get on with it. They know exactly what to do. And I was so happy to see how everything come together with the case study by Carlos, and how everything is put together through the decision support system. So that all of that to me is very interesting. And then we have this, whether you like to call it a bonus or not, we have COVID-19, which then challenge all of us. And in a way, COVID-19 is a case study but it's not a case study we choose. It was a case study that dropped on our lap and we had to react to it. And as I expected, you guys has all the expertise and the originality and the innovation to contribute to that. And I think that is something that I would like the Commission to be aware, or at least the Nanosafety cluster to make it very clear to the Commission that the Nanosafety cluster has what it takes. I'm not just speaking about BIORIMA alone, yeah? I'm talking about all our other colleagues in other projects, because in a way, we're not very different from anyone else, yeah? That message has to be taken forward, that the nanosafety community deserves to have a say in the policy, in the strategy to combat viruses. I think that is something through Andreas or other, we have to convey this message, is that we're not just another group of scientists looking for something to do. We have the expertise, and you can hear it through how fluently and with ease people like Marco or Miguel or Ben talk about it, or Terry. You can see that this is something that really we should be put in the forefront, and hopefully through the nanosafety cluster we'll be able to make our voice heard. Okay, that is my personal view as a coordinator. I think this actually will set the theme for the coming call, is that I think we should understand that nanobiomaterial or engineered nanomaterial, the division is artificial, that deep down, both these materials cross the border all the time. And we have plenty of people with expertise from nanosafety that can contribute to nanomedicine and treatment and the development of drug treatment and so on. Yeah.
Thank you, Lang. I think you are perfectly pushing the button. At the one hand, also when I follow a little bit the comments here in the chat, that exactly this collaboration is definitely one of the key assets we do have already in this nanosafety community. May I ask now, Susanne, to introduce the second question shortly with the...
Yes, actually, most people are really busy with the second question, because I think everybody's keen on adding sticky notes, which is perfectly fine. So we can close definitely the first question. I think everybody had the chance to add a sticky note with the name and organization. So I started to tidy it up so that it's readable. So for the second question, it's called collaboration offer. So it's really meant that everyone can add the specific expertise that they would like to offer in a potential future collaboration. I already see quite many sticky notes and input here. I will also start to tidy up a little bit. Maybe we can give 3 more minutes for that topic before we move to the next task.
Indeed, I just see, Lang, you might be still active in your post-it just in the first section. If so, you may just close that and make a new one. Anyhow, some of you, I just saw that you have put maybe only the pre-name, the first name into the box. You might come back to your personal post-it throughout this time to add maybe your full name and affiliation, if you like to. Looking into the already positioned topics, I think we do have really a lot of important expertise already listed here now. It will be, I mean, it's amazing how active you all are now. And it's really great. I just want to kindly ask you that you try, even if it's maybe at the moment not easy to read, to put your post-it directly into exactly that poster part where you want to have it at the end. It seems that some of you have put it in between some of the topics, which might make it a little bit tricky. Anyhow,
they can zoom. People can zoom so they can see it easier.
Yes, exactly, because there are several functions we would have, and at the end we will of course sort out to have a real nice overview what collaboration offers are there. Anyhow, collaboration is always about topics, about output, and about that at the end someone should have a benefit out of our work. And of course, one of our duties is that all the society, the taxpayers around us do have at the end a benefit from the work we deliver. I think BIORIMA made a really great example, and I hope the Commission also took note about the outputs, how important these aspects were tackled in this project. I think that shall definitely also feed into the future collaboration topics that might be called for or might be tackled by these groups in smaller groups, in smaller consortia. So if you are thinking about the collaboration on the one hand, also please think and start to think now for also the next question, what would be these special topics? What would be these special research items you see relevant to tackle in the near future? Oh, that's like in school. Thank you, Susanne.
Yes, it was the end of task 2, and now everyone is invited to start adding or continue adding actually inputs to the third one, which is really about future trends, current needs, what's based on all the outcomes and the interesting results, what we heard from BIORIMA, what could be done next, what should be done next, and what's the way forward in the future.
Exactly, and as Susanne already said, that's following what Bengt and Lang and colleagues discussed in the sessions before. There is something which can be delivered by our groups here. So please try to be a little bit visionary on that point, not only about, let's say, the specific expertise that hopefully you have already put into the previous section, but also thinking forward. What do you think is the message can be delivered to which kind of topics? What kind of needs do you see that should be tackled? Because at the end, we, of course, will be measured about our output, about the impact we generate. And I think, especially to know what gaps are there, will support us in the future visionary thinking about what impact we can deliver with having the nanobiomaterials and the nanosafety very, very close to each other.
Can I say something? Sorry, Andreas.
Lang, I'll ask after. Go on, Lang, please.
Okay, Andreas, I think of there's one cross-cutting issue across all our projects, which I think we lack a vision and a strategy, okay? And that is the communication. I think I agree with everybody say about all the scientific stuff. I mean, I'm a scientist myself, that's not the issue. Now, we are all in nanosafety, and BIORIMA is basically a nanosafety for material used in medicine, but it's basically nanosafety, okay? And all our project has a work package on this on dissemination and communication. You agree? All of us, okay? But I'm not criticizing anybody. BIORIMA too. We done a good dissemination work packages, but the words that we disseminate our idea is a very small world. Even when we're talking to representative of industry or NGO or whatever, they are all scientists like us. Now, the worst thing that ever happened is we agree to disagree, and that's the end of the story. Okay, but I think if I can remind you guys back a few years, I remember, Ben, you are with us already then, right? The CNR was the first bunch of people who tried to engage the issue of nanosafety with the general public in France. You remember the time they set up what they call the Grenelle, and this really stretched back the memory a bit, okay? But those of us who were there, the result was a disaster. It was a serious disaster that eventually, very shortly, the CNR closed the dialogue with the general public because the reaction of the French, whoever, the people who participate to the dialogue about nanosafety was so aggressive that it was buried. I remember that. I talked to Oliver the other day and I asked him, did he remember it? And he confirmed it to me. That was, looking back with hindsight, that was the first taste about communication with the public is like. And we had a really interesting encounter with the public. Now, at the moment, as you all know, from the scientific governmental side, the communication strategy doesn't exist. It doesn't really exist. To make it a point, when President Emmanuel Macron was asked, how do you persuade the anti-vax to get vaccinated? His answer, and I quote the French word, is to emmerder them, to really emmerder them to the point that they had so fed up that they go and get the vaccine. Now, is that the communication strategy that we're going to follow in all our work packages to emmerder the public until they agree with us? Now, I don't think that is the right way to do, but that really illustrates the point. The point is there is no communication strategy. We are keeping to our comfort zone. And
definitely, Lang. May I give the word to Janeck and then to Terry, please?
I just want to say that when you get all these brainstorming, very nice. I like the sessions, very nice session. But getting all this brainstorming, you want to make them more concrete. Please contact either Bengt or myself to make a paper on it, and then it could be even more concrete. Then you could do that, right? Bengt has a very nice Frontiers journal, and we have a special issue. So.
Perfect. Thanks, Janeck. That's perfectly also for the fourth question now. Terry, please, Terry, keep coming.
This is a quick one. I've got a more positive experience of communication, which at the time didn't feel very, very positive, but it was. And I'm wondering how well it would translate to the modern era with social media. I was invited by industry to make a presentation to MEPs in the European Parliament in 2011. And before I went in, I was lobbied by the big companies, for God's sake, don't talk about the safety of nanoparticles. But, well, I've got to be honest, I knew there would be very powerful green NGO lobbyists who will be there throwing rocks at me, and they were. But I decided beforehand to put in my talk a presentation about all the good work that the nanosafety cluster and you guys were all doing to show that something positive was going on to protect them in the future, that the environment, health and safety issues were not very well marketed or understood in those days. So I described a strategy leading through to safe by design, how you could do safe by design. And and at the end of my presentation, a slide many of you have seen before, I showed what's inside my eyeballs after retinal detachment and subsequent damage to the hard lens in my eye. And I now have concentric dual function lenses in my eye. There's a nanostructure diffraction grating and on a hemispherical lens, and showing that I'd had these in my eyes for 2 years. They're still there, they're still working. And the mood of the meeting changed. The big companies got more open and they stood up and supported what I said. And the audience turned on the green NGOs. They thought their behavior was not right. They were not working on the basis of the science. So we got to get the science communicated to them in an attractive and understandable way. We've seen this in our own country here, when we've had good scientists stand up and explain how the COVID-19 vaccine, how the COVID-19 crisis is going to be managed and what we've all got to do. So I think just keep at it, keep it relevant.
Thank you, Terry. I would say that's perfect also in the face of our mural exercise, of our whiteboard exercise now. I just wanted to double the words that you really made already a great effort here on communicating things in this project also to the broad public. We have now this fourth question here now, the public communication on the safety of nanobiomaterials. Of course, there are some challenges, and there you have already mentioned now again some points that shall just feed into that. And I really like this comment from Janeck about making a joint publication out of this to make a clear statement of maybe the challenges, the gaps, and the chances we do have with the public communication on these scientific points. Lang and our team, when we prepared this session, were already discussing about maybe there are some volunteers in this BIORIMA consortium about going for a joint publication to make a statement on the public communication. And this was so for our fourth question, which Susanne has put into this whiteboard, is that we want on the one hand get your visions on, okay, what challenges to achieve so-called quality communication. It's maybe those who have already contributed to the INIS Nano initiative know already this terminology, which came up from a very wise guy we had seen for years in our community. Georgios Katolagranakis is still one of those who will never end with work on making things happen. So the credits for this terminology shall be on his hand. I think quality communication of scientific experts could make a difference. I would be super happy if we come to the conclusion after this meeting to go for such a publication, which takes more or less this big case study, how Lang called it a little bit earlier, proving that the nanosafety community has contributed a lot to make things happen. I stop here. Gary, I saw that you have raised your hand, please.
Andreas, thank you. Gary Hutchison from Edinburgh Napier. I fully support what you're suggesting there about a publication, and it's just to make everyone aware. Last year, DEFRA in the UK's Hazardous Substances Advisory Committee, we did a Delphi-style review with an international panel looking at chemicals and the challenges in the future for us to address in relation to the Green Deal, but also in relation to what kind of challenges will toxicologists and the public have to face. We've written this up. It will be a publication that will come out. It will be submitted hopefully next month. But it's a really nice summary of a panel's review of the major challenges and major threats. Nano appears within that, as does the advanced materials topic, and also chemicals that are being used in different areas, and it crosses human and the environment. So this has been written in such a way to show the public what's on the horizon and perhaps the community and trying to marshal regulatory bodies and governments to coordinate activity across the world to try and focus on these key challenges. Really interesting idea. It was a really good process to be part of, and I think something focusing on nano could emulate that quite nicely. So I just wanted to share some enthusiasm for it and say that I've seen it work quite well in other areas and broader chemicals. Thank you.
Thanks.
Yeah, thanks. I want to follow up on what Lang was saying earlier. I mean that if we try to evaluate really what we are doing, we seem to be preaching to the choir, but in fact, if we want to reach the general public, this is really a totally different issue, and I'm certainly not against joint publications, but we will reach people who read publications in the scientific literature. I think we I mean, I have spoken to so many journalists over the years, and it's quite staggering the lack of knowledge among so-called science journalists. So one shouldn't underestimate how challenging it is to get the message across. I think people can sort of digest a tweet, but not a paper. And maybe one should try something different. Maybe we should engage with the cartoonist and do a comic strip on nano, because really I think we have to find new ways to communicate. I applaud all these initiatives, but it is something that we have seen during the pandemic, and which I guess this general public has also seen, is that even scientific experts cannot agree. We as scientists know that this is part of science, but the general public, I think, still doesn't get it. They don't understand why we can't agree on the virus and on the vaccine and so on. So we have to make it very simple, I think.
Bengt, I do share your comments to a certain extent. I'm definitely clear that there is a different kind of communication needed. I'm not sure how simple it's possible or useful, because on the one hand, of course, it's possible to make cartoons and things like that. But on the other hand, it's not possible to interact then with the people who might be the visitors looking at such a cartoon. What I saw now, I mean, all around we have these discussions, and you can think about all these measures, whatever you want, but there is no scientific-based dialogue ongoing. It's more an interaction of opinions, of interpretations, but not about real facts. And I think if it's possible to find a way to communicate real facts based on scientific results in a way that it's maybe a kind of mixture of this internal scientific community communication, which is the publication, but has elements which can then be used for tailoring the broad public, I think then it would be a step forward. But I'm not sure if it's even possible to publish such a paper in any kind of journal, because it might be rejected, you know.
It's very quickly sorry to interrupt, just that everybody is aware that the fourth question is divided. So on the upper part, you can add input to this question: what are the challenges to achieve this quality communication? And the lower part is really meant to collect names who is interested in such an activity that could be continued by the Nanosafety cluster. So if you would like to participate in such an activity, please add your name also to the lower part and put other comments in the upper part. Sorry, thank you.
Thank you, Susanne. I have now sent Lisa first and then Janeck. Please, Lisa.
Yes, I agree that we must be clear on the fact that dissemination is totally different than communication, of course. It has a different language and a different objective. So communication is what we're talking about when we talk to people. But I think that there is no tool for communication between, that relates scientists to people, because scientists have a mindset. If you explain a fact to a scientist, like to an engineer, he will understand it. But if people don't have this training, the majority of people don't look at the facts the same way as scientists and engineers do. So you see that in the new Horizon Europe topics, there is this very important part, which is even more important now than ever. And I think that the European Commission is very visionary on this, which is the engagement of social science and humanities. You need to engage people who are in the social science and humanities. You need to engage sociologists, for example, who can really know how to ask questions. But I think that even trying to be looking more forward than this, I think that we need a new type of expertise. We would need to train younger people, I think, to be both experts in sociology and science, at least understand both worlds, because you cannot be probably expert in both of them, but be pretty good at understanding really both sciences, the sociologists, so the social sciences and the scientific scientists. I think this type of people could be really the tool that we can use to make citizens understand science and not be afraid of it, because when people don't understand something, don't know it, they are naturally afraid of it. So I think there's no easy tool, Lang. There is no chat box. So I don't think there is a tool now. I think we need to be looking forward and thinking what is probably a new expertise that is needed.
Let Janeck say, and then I comment. Go.
Thank you. I think that's all true, what has been said before. I just want to also comment in relation to this and to Bengt. I think first we do a scientific paper among ourselves, something like one or many, and then we disagree and agree on what we write. And I think it's perfectly okay to disagree and write that in a paper and so on. I think that's probably a way forward. Then from there we can extract to the general public. I just have one thing, when I leave this Zoom meeting and then I have to go shopping or whatever I have to do, right? I'm a normal citizen and I go down and then I buy whatever or interact based on facts. I think most people think they argue also outside science based on fact, even though we think that their facts are wrong. It's from Facebook, whatever it is, but they still argue that they are true. So I think we can catch them up in making, what we don't need to run into. I mean, sorry, I'm speaking out of my field, so just take me with a grain of salt, okay? But we are not speaking to a total stranger. We're not speaking to somebody from another planet. They use arguments. We just need to get more scientific arguments into their arguments. And then I think it would be better. And it's a long haul. I think it's a long haul. And I don't think there's anything, I don't think we need to bend into anything. I mean, scientific arguments are scientific arguments. They're the best way forward.
Thanks. Can I say something, Andreas? Can I add to the, okay. Lisa, I agree with you totally. And I think Lisa highlights this point very clearly, is that we haven't got the tools and the expertise to really address this group of stakeholders. And I think this is the message that we must convey. And maybe the best way to convey is to get all the people who run the work package and dissemination across all the projects to sit together and say that, look, we've been working within our comfort zone, using the comfort tools that we have, such as writing paper or whatever, which is something that we feel at home with. But the few occasions in which we really have to face the public, it has left us wounded and fearful when we talk to them. That's my impression. And I think we have to somehow, just because it is difficult, as Lisa said, it doesn't mean we should give up on it. I think we have no choice, because this could scoop everything. And that's my first point. The second point I want is a message of hope, is that if you look at the anti-vaccination group, right? I read it somewhere that there's only 12 of them in the planet. 12 people who managed to motivate millions of people. And we are a community, boys and girls, and we somehow are fearful of them. There's only 12 of them. They're not even that organized. And they have no strategy, no funding, no money. And somehow they motivate millions of people. So they must have done something right. So that is something that I really want to put in perspective in there, is that we got what it takes. We were just barking at the wrong tree, that's all. So please, now is the time to face up with the problem. And the third thing I want to say, and this has been touched upon by Lisa, is that you have to understand, you have to profile the public. You have to understand what is it that behind them. Because from what I understand, from my perspective as a citizen of the United Kingdom, is that there's a lot of uncertainty and fear in the mind of the public. And that is really what drives the skepticism, okay? That the fear of science is in fact a symptom of some more deep-rooted fear. And these people, these 12 people, just know how to capitalize on it. So when we talk to this stakeholder, unlike our fellow scientists across the board, who has a rational frame of mind, you have to understand that we are trying to communicate something with people who are basically fearful. And that anxiety is very palatable when you talk to these people. You can easily see behind them and see this fear. Because for me, as a British, I saw it very clear that while not all the people who vote for Brexit are actually anti-vax, but I can tell you one for sure, that all the British anti-vax vote for Brexit. It is something much more fundamental. It's something fearful of, they call us the elites, they call us whatever, the deep state or whatever word. And that is something that we have to address. That there is a large swath of people in this society who are very fearful and anxious about the future and the world. And we have to face it up if we are going to talk to them meaningfully. Okay?
Thank you, Lang.
That's my view.
Thank you very much, Lang. I think we are now on the final minutes of the whiteboard exercise. I see that still there are some people are running around in the mural. Maybe you can come to a conclusion for yourself that we then can close that part and that we can digest afterwards the things. I also have put, just for security reasons, that we don't miss out the one or other volunteer, into the chat, the email addresses of Susanne and me, that we just try to interconnect the next days then about the potential publications and so on. And of course, it will be one of our ideas to step forward with nanosafety cluster activity on that, because there is already a community existing that can be used for exactly these things. And communication definitely should be one of the topics in the center of such a cluster. This brings me more or less to an announcement. And Susanne, if you're able to share your screen for one more minute, that would be really perfect. The colleagues from several projects and involved in this is the Nanotechnology Industry Association. So NIA, the most will know that. Will drive, starting at 3 p.m. Central European Time, a communicating about nanomaterials webinar. So those who are not already fed up of sitting in front of their screens might use then the 15 minutes break after Lang has closed the BIORIMA meeting for recovering, and then join on a last minute spot this nice webinar. It's perfectly in the same direction. So we have a perfect, let's say, bridge from BIORIMA, high scientific stuff, over the public communication activity we just had now towards that webinar. The agenda is presented here on the screen, so it's not too long. It's 1.5 hours, more or less. And I'm quite sure that you will like what you will hear there, and that it's also a possibility to get another bunch of interested people for this kind of real joint effort in the nanosafety community. With this, I'm finally closing this part of the session. And thank you very much for your very, very active contributions. With this, Lang, I give back the floor of the 7 last minutes of BioRima final event, that you can close the meeting and and hopefully see the one or the other later on in the following webinar at 3 p.m. Thank you very much.
So this is it. It's we finally finished BioRima. Where if you add in the extra 6 months that we have for extension, it's a 4.5 years. I got a mail from Gary Hutchison, who wrote to me and he said, I couldn't believe it's a 4 years project. 4 years gone by. I correct him. It's actually 4.5 years. And it went like a tick. It has been, of course, I hope I speak for everybody. It was a lovely time to work together. I had wished that the COVID wasn't around and we could have met regularly throughout the time that we were together. I had a lot of plan about it, such as the celebration for the Nanosafety training school, which we managed to drag it all the way to its 10th year. Unfortunately, it was cancelled because of COVID. But me and Suzanne, and others, we have a great plan about to throw a massive party, and you can see it here. So we are plotting here to have the plan. But one thing about life is that if you do not achieve something, usually that's the way life tells you that you have to try again. So we didn't have what we planned to do, Suzanne. That only prompted me to work harder to make it happen. So I hope I'll be around to work with you in the next proposal and call. And as you see, the world is full of challenge. One thing we learned from working together through BioRima is that reality will always hit you where you least expected it. In a way, the COVID story came, but it took a while for us to realize that, in fact, it required all our expertise. You know, we never expected it. The Commission never expected it. They thought of, let's get the vaccine and we get it over with. But life is more complicated than that. First, you get the vaccine, and then there are people who object to it. And then they ask the question of safety of material, and all this and that. And I think that's the first thing. And the second thing is that it will take a case studies like COVID to realize that we have what it takes to answer this thing, that we are a community with the right expertise. Yeah. I mean, you listen to all the talk so far, and you see the ease and the ideas that come from all our scientists here shows that they can jump into this area of COVID vaccine without a great learning curve at all. It is the same thing that we can address, and throughout our nanosafety communities, there are plenty of authors as well. And I think that I think that was a wake-up call, is that we cannot be so focused as to forget that we have plenty of expertise, and our expertise are very, very useful. Okay, very useful, very timely, very innovative. Okay, and then the last comment I have before we close is that it would take an event like the COVID-19 to make us realize that we live in our little comfort zone. All our communication strategy and everything, they work fine. And of course they work fine within the community that we dwell in. It's fine. But the world is bigger than our community. And I think it's a message that I hope, Andreas, you will carry it to the entire community and say that you cannot remain so complacent about talking to each other and publish papers. I mean, papers are fine, no problem, but the public doesn't read paper, as Ben pointed out quite nicely. They don't read papers. The tools of communication are elsewhere. And my last thing before I say that we have no choice to address this issue. However, we on behalf of everybody, I would like to say also that we have to be careful. That world that we think that we have to engage is not so simple. It's a very complicated world outside our little bubble. And it could mean our security be challenged. It would mean people recognize Susan in the street and start picking on her. You know, it could mean any of these things. You know how it happened. But however, the view is we have no choice but having to face up to it, because it is our world. It's about scientists versus the world. And I think we have to take it as a challenge. So on my few ideas that I share with you, I think thank you all of you, and thank you Marcello for being such a supportive and interesting party to this endeavor. And I hope that there will be BIORIMA 3. We will think of something and we will make the Commission proud of what we have done. Just as in BIORIMA, we are proud of what we have done, and I'm sure the Commission will be too. So thank you, everybody. Thank you, Marcello, and I look forward to the review meeting in Brussels. Thank you. Okay, we can close now.
Thank you. Yes.
Thank you. You want to say something, Lisa?
Yes, please. All presenters send me the presentations of today, please. It's just okay. Hope to see you soon. Maybe next time should be really into rain, as this time. Too bad. Okay. Thank you.
Sure.
Thank you.
Thank you,
everybody.
Thank you.
Bye.
Bye.
Bye. Hi.
Ciao.
Bye.
Bye. Take care.
Hope to
Bye.
see you soon.
Bye.
Bye, everybody. Thank you.
Anna?
Ecco, aspetta, te l'avevo mandata qui, Lisa.
Dove l'avevi
Ti avevo mandato
messa?
la presentazione qui. No, ma si sta caricando perché è un po' grossa, se no te la mando per email.
Sì, dai,
se vuoi
mandamela per
chiudere.
email. Sì, anche perché qua
Dai.
sto registrando e non vorrei che...
Ok, va bene.
Così
D'accordo. Ciao.
Ok, ciao.
Ciao
Grazie.
bella.
Ciao, ciao. Hildegard. Still here.
Ecco, aspetta che esco. Ecco.
Ciao Hildegard, I registered and recorded, and also Stefania did record, so hopefully we didn't miss it.
Okay, so that the recording will be converted normally at